Hello, everyone, and welcome. On behalf of the Duke Margolis Institute for Health Policy, I'd like to welcome everyone to today's virtual private roundtable, Advancing Biomarker Use and Regulatory Review, Evidence, Engagement, and Ecosystem Opportunities. I'm Valerie Parker. I'm an Assistant Policy Research Director for Regulatory and R&D with Duke Margolis, and I'll also be serving as one of our moderators for today. Thank you for joining us for this discussion about improving how biomarkers and surrogate endpoints support regulatory decision-making, especially in biomarker-supported accelerated approval and rare disease contexts. Today's public workshop is one component of a longer-term research project to develop recommendations and a framework to facilitate biomarker development and use for regulatory decision-making. Our goal is to identify practical policies, stakeholder recommendations, and infrastructure needs to support an environment where science and regulation can improve biomarkers. Based on stakeholder interviews and feedback sessions, a literature review, and an expert roundtable, Duke Margolis has developed a preliminary policy roadmap organized around 4 goals, which my colleague Nicholas will present next. During today's public workshop, Duke Margolis will solicit expert and audience feedback on the feasibility, utility, relevance, and prioritization of the recommendations included in the preliminary roadmap. To do this, we'll review the roadmap and then hear 2 use case presentations that will ground the workshop in concrete examples of biomarker development and regulatory use. Next, we'll then have a session, we'll have 3 sessions focused on different aspects of the preliminary policy roadmap. The first will look at evidentiary expectations for biomarkers' intended regulatory use. The second session will examine shared infrastructure, incentives, and evidence reuse mechanisms to support biomarker development. And the third will focus in on opportunities to improve the transparency, consistency, and accountability of biomarker-related review and engagement across the development lifecycle. We'll then close the day with a panel discussion that considers key themes, audience feedback, and areas of convergence or remaining uncertainty from the workshop discussions. We'll have until around 4:30 p.m. Eastern Time, and I'd like to encourage each of you to join the conversation today. Before we get started, I'll review some brief logistics. This workshop is being recorded and will be published to our website within a few days. The audience Q&A is intended to facilitate an open and dynamic discussion. And if you're online and would like to participate, please submit your questions through Slido, which you can access using the QR code or link, which we will post in the Zoom chat. If you're attending in person, you can also scan this QR code to submit your questions through Slido or line up behind a microphone, which we will turn on during the Q&A sessions. In addition, the audience will have opportunities to engage through a series of 5 audience polls, which we will open at the top of each session's discussion. Please be prepared to participate in these, as the results will be incorporated into our discussions after each of our session's panelists have had the chance to share their insights. And next, I'd like to go over our Statement of Independence. Our academic statement of independence means that neither Duke University nor Duke Margolis Institute take partisan positions, but individual members are free to speak their minds and express their opinions regarding important issues. This project is sponsored by the Biotechnology Innovation Organization, or BIO, although Duke Margolis retains academic and editorial independence. Now, to get us started, I'd like to welcome my colleague Nicholas Harrison to present an overview of our preliminary policy roadmap and research.
Thanks, Al. I'm Nicholas Harrison. I'm with Duke Margolis, and I'll take the next 15 or so minutes to introduce the workshop, orient us to the policy roadmap, and talk a little bit about what we hope to get out of today. As you all know, biomarkers can make drug development more efficient by helping sponsors to generate evidence earlier, design more feasible trials, and support regulatory decisions when traditional clinical endpoints are delayed, difficult to measure, or simply impractical. Those opportunities can be especially important in rare, serious, and slowly progressive diseases where small populations and long disease courses can make conventional development challenging. At the same time, External stakeholders have described difficulty planning how biomarker evidence should mature, how analytical and clinical evidence should come together, and what may be needed for increasingly consequential regulatory uses. That does not mean one evidentiary standard should apply across every biomarker or context of use. Biomarker evaluation is necessarily context dependent. The questions for this workshop is whether expectations, feedback, and development pathways can be made more understandable and actionable without sacrificing that flexibility. Our entire project's goal is to develop actionable policy recommendations and a roadmap that can improve biomarker development and regulatory utility. Our primary focus is biomarker evidence considered within individual product submissions, particularly biomarkers used as surrogate endpoints or reasonably likely surrogate endpoints in accelerated approval and rare disease contexts. The FDA's formal biomarker qualification program is related and remains important, but it is not the primary focus of this workshop and the roadmap that we're designing. We are concentrating on practical policy opportunities and product-specific regulatory review. The roadmap draws on policy research and stakeholder input. It should not be read as an FDA position or as an assumption that external perceptions necessarily reflect how the FDA understands its own processes. Our policy research has been an iterative process. In the first phase, we combined a literature review, stakeholder interviews, and a broader landscape review, and then used those inputs to frame an expert roundtable. In the second phase, we analyzed that roundtable, held additional feedback sessions, and translated the recurring issues and proposed solutions into a preliminary roadmap. Today's public workshop is the next major test of that draft. In the third phase, we will analyze this workshop and survey input, revise the roadmap, and prepare it for publication. These inputs help us triangulate themes, but they do not imply uniform agreement. The roadmap is preliminary because today's discussion is intended to challenge its assumptions, identify gaps, and distinguish ideas from feasible policy opportunities. Our central finding thus far is that scientific discovery is only one part of the challenge. Stakeholders also have to translate biomarker evidence into fit-for-purpose development strategies, ask regulatory questions that can produce actionable feedback, and build infrastructure that supports evidence generation and reuse. First, stakeholders described evidentiary expectations as difficult to operationalize across different biomarker uses. This may in part reflect the legitimate fact that evidence needed for something like an exploratory biomarker is different from the evidence needed for a biomarker-based surrogate endpoint. Second, analytical validation and clinical validation often develop on different timelines. Programs need ways to preserve evidence continuity as assays change or biomarkers move into a new setting. Third, stakeholders want more predictable planning tools, while the FDA review necessarily remains context-specific and evidence-dependent. The opportunity is not to eliminate scientific judgment, but to help stakeholders prepare for it more effectively. Finally, shared evidence generation may be necessary for difficult biomarkers, but it can be hard to coordinate, fund, govern, and reuse. Those four findings provide the bridge to the roadmap. The recommendations are organized under the four policy goals, but they are also tagged by where they apply across biomarker development and use: discovery, measurement, and regulatory and development use. Those tags matter because a recommendation that is useful during biomarker identification may not be the right tool for assay bridging, endpoint selection, or regulatory review. They help us ask which actor should do what and at what stage. The roadmap is intentionally preliminary. Today, we need to narrow the recommendations, prioritize them, assign realistic roles, and identify credible implementation pathways. With that structure in mind, I will briefly walk through each goal. Goal 1 is about operationalizing fit-for-purpose evidence development as biomarkers, assays, and intended uses mature. The preliminary recommendations explore tiered evidentiary development, context-dependent integration of evidence, and clear expectations for assay readiness and evidence continuity. A useful framework might show how the strength and type of evidence should evolve with the intended use, the consequences of the decision, and the maturity of the measurement approach. The intent is not to create a universal checklist or a single evidentiary threshold. It is to give sponsors, researchers, and reviewers a more practical way to plan, identify evidence gaps, and explain why a particular package is fit for a particular purpose. Goal 2 focuses on how stakeholders learn from complex, context-dependent biomarker review and carry those lessons into future development strategies. The preliminary recommendations consider public-facing case examples, clearer descriptions of recurring evidentiary considerations, and communication about how prior evidence may inform related programs. This is not a generalized critique of FDA review, and it does not assume that a prior decision should determine a later one. Public materials would need to preserve the FDA's flexibility, protect confidential information, and explain the limits of any analogy. The workshop can help identify which products would actually be useful. For example, case studies, explanatory materials, or cross-cutting review learnings, and which actors could feasibly develop and maintain them. Goal 3 addresses timing. Sponsors often make important assay, endpoint, and evidence generation decisions before the full regulatory strategy is clear, and changes made later can be expensive or impractical. The roadmap therefore asks whether earlier, structured feedback at key milestones could better align evidence plans with the questions likely to matter later in development. This does not necessarily require a new meeting type or imply that the FDA can resolve uncertainty before the evidence exists. The workshop should examine what can be accomplished through existing pathways, what information sponsors need to bring forward, and when cross-functional engagement would add value. The practical objective is earlier identification of pivotal questions and fewer avoidable disconnects across the development lifecycle. Goal 4 turns to the ecosystem. Early biomarker development can require substantial investment before regulatory value is established, leaving first movers to bear costs that may eventually benefit an entire field. The preliminary recommendations explore shared investment in early evidence, common data and measurement standards, and governance models that allow responsible evidence reuse. Shared infrastructure does not mean that all data must be open or that evidence will transfer automatically across diseases, assays, or contexts of use. Consent, privacy, intellectual property, data quality, and comparability all set real boundaries. The workshop should help clarify where collaboration is genuinely pre-competitive, which organizations are positioned to lead it, and what sustainable investment models could make shared work possible. Across all 4 goals, we need today's discussion to answer 3 practical questions. First is prioritization. Which recommendations could have the greatest impact, which are feasible, and which need to be narrowed or set aside? A strong idea is not enough if the actor, evidence, resources, or implementation path is unclear. Second is responsibility. Which opportunities are appropriately FDA-led and which depend on sponsors, consortia, patient communities, researchers, clinicians, or neutral conveners? Many of the challenges are shared, so the roadmap should not place every solution on a single institution. Third is implementation. What concrete products would make a recommendation actionable? That might include pilots, templates, maturity frameworks, case examples, shared standards, or infrastructure with clear governance. We also want to hear where the roadmap has framed the problem incorrectly, where legitimate constraints limit the available options, and where uncertainty should remain visible rather than being forced into false consensus. Those distinctions will be essential when we revise. The workshop's 4 sessions are designed to evaluate the roadmap in progressively more concrete terms. We will ask panelists in the audience about the feasibility, relevance, and priority of the preliminary recommendations now on the table. Throughout the workshop, we will use Slido for audience polling and Q&A. Each polling exercise presents the policy recommendations for goals 1 through 4 and invites participants to identify 3 to 5 policy recommendations most worthy of additional consideration. In our last session, we will review preliminary findings from the polling in addition to inviting you to rank the importance of the goals themselves. We also want to surface additional policy ideas, implementation consideration, and areas of both convergence and continuing uncertainty. Disagreement will be useful if it helps us understand where a recommendation needs a narrower scope, a different actor, or more evidence. Today's discussion will inform a more complete, prioritized, and actionable roadmap, but it will not make the roadmap final. We will analyze the workshop and audience polling input, revise the recommendations, and continue to assess feasibility and implementation. As you participate, I encourage you to focus not only on what should change, but on who could act, through what mechanism, and with what safeguards. That level of specificity is what will help turn a preliminary policy roadmap into a useful one. With that, let us move into the next part of today's workshop. Before we begin the panel discussions, we want to ground the workshop in examples of how biomarker evidence is developed and ultimately considered for regulatory use. These presentations will help us surface practical questions about evidence maturation, stakeholder coordination, and how lessons from one development program may inform broader policy opportunities. We'll first hear from Nicole Gormley, who will discuss minimal residual disease and multiple myeloma, followed by Shraboni Sharkar, who will present on the Neuro Toolkit. We encourage you to keep these examples in mind as we move into the workshop's broader discussions of the preliminary policy roadmap. Nicole is the founder of Gormley Advisory, an independent regulatory consulting practice focused on oncology drug development. During her nearly 15 years at the FDA, Dr. Gormley oversaw drug development for multiple myeloma, lymphomas, and chronic lymphocytic leukemia, and provided scientific and policy leadership on early endpoint development in oncology. She was instrumental in advancing novel endpoints, including minimal residual disease, and championed efforts to improve clinical trial representation. Thanks for joining us, Nicole.
Great. Thank you so much to the organizers for the opportunity to share a little bit about minimal residual disease and multiple myeloma. And so specifically, minimal residual disease was a biomarker that was developed in consultation with the FDA and specifically the review division there at the FDA. And it was development of minimal residual disease to serve as an endpoint that could support accelerated approval in multiple myeloma. And so basically, I'm going to share a little bit about the story, the why of minimal residual disease and multiple myeloma, how we got there, and some of the next steps that still exist. So why? Over the past 20 years, there's been approval of multiple agents in multiple myeloma, more than 20 in about 20 years exactly. And this has really been unprecedented growth in this field. These are very highly effective therapies that have resulted in significant improvements in clinical outcomes in this disease space, such that it's now thought that individuals diagnosed with newly diagnosed with multiple myeloma with standard risk disease have expectations for overall survival that are greater than 10 years. So really significant improvements in outcomes in this disease space. But along with that comes several challenges. These therapies are very highly effective, and in the newly diagnosed setting, when used in combinations, we're seeing response rates that are greater than 90%. And even in the relapse refractory setting, with therapies, when they're used in combination or even as monotherapy with some of these agents, they're very high response rates, and that can make differentiation somewhat challenging when trying to establish the outcomes and the safety and efficacy of new therapies benchmarked against prior therapies. So there was really an interest in the field in having other response and assessment measurements that could be used other than just response rates and other than the long-term outcomes of progression-free survival and overall survival. Continued development in this space. So that's really the why of minimal residual disease and multiple myeloma. Overall response rate had been used long-standing history of using overall response rate to support accelerated approval. And then the readouts for long-term outcomes of progression-free survival and overall survival are really too long to be relied upon for further drug development. The high response rates that I showed you that are seen with these novel therapies really sort of limit the ability to discriminate between therapies of existing therapies and novel therapies. And so there really was a need to have other therapies that would allow for greater differentiation. And so this really sort of led to a situation where there was almost a clear unmet need for a novel endpoint. And that's not something that we talk about a lot, but it's something I think it's really pivotal in this unique situation. So how did we get there? So I'm going to share that there was a lot of community involvement across the ecosphere as it relates to development of minimal residual disease. In 2013 to 2014, the FDA held a series of workshops to discuss use of minimal residual disease in various heme malignancies. These included ALL, AML, CLL, and also multiple myeloma. And really, these workshops were to discuss potential response biomarkers and next steps for further development. In the academic community, Dr. Ola Langren had the first MRD and multiple myeloma annual meeting, and this was a meeting that was held annually at Memorial Sloan Kettering and later at University of Miami, really focused just on minimal residual disease and multiple myeloma, with discussion of novel assays, latest advancements and technologies, and how to best incorporate MRD clinically and in drug development. And then I'll just lastly mention the 2017 CCR publication, which was a collaboration initiated by an FNIH Biomarker Consortium, which really sought to clarify the role of minimal residual disease in patient care and drug development. But these efforts really point to large community involvement across multiple spheres, regulators, academia, clinicians, industry, patient advocacy groups, really coming together in multiple areas to try to advance use of minimal residual disease, given the need that's been demonstrated. And so in 2016, as a result of many of these efforts, the International Myeloma Working Group, the IMWG, updated their response criteria, specifically to include assessment of MRD as one of the response assessments. The criteria stated specifically that MRD could be assessed via different technology platforms, whether that be flow cytometry, next-gen sequencing, but it specifically established a minimum threshold of 10 to the minus 5, regardless of the testing modality that would be used to determine what MRD negativity was. And these efforts really had, as it relates to collection, assessment, reporting of MRD, really sort of led to standardization and harmonization within the field, such that then going forward after that point, clinical trials across the board, when they were assessing MRD, whether as an exploratory endpoint or a secondary endpoint, included MRD using this one established benchmark. Subsequently, after that, in 2018, the FDA issued draft guidance on minimal residual disease in heme malignancies and really discussed the regulatory uses of MRD. It provided biomarker definitions using the best criteria and also really laid out some of the considerations for how to develop MRD as a potential endpoint. This included the discussion of the surrogate endpoint acceptance or qualification pathway, meta-analyses and meta-analytical approaches to validate a potential biomarker as an endpoint. It talked about use and inclusion of MRD in clinical trials or for patient selection, patient enrichment. There was a discussion regarding the technology and various considerations of different assays that were used, and then also disease-specific considerations for the different heme malignancies and some of the specific considerations that come into play when you're thinking about in the heme space, different disease considerations regarding location of disease or other factors. And then also ultimately also included information on regulatory submissions which used MRD. So this was also helpful, I think, at the time to sort of display that the agency had interest in development of MRD. It also provided guidelines and sort of some criteria about how to use MRD in drug development, and really sort of started as well some of the initial work on developing MRD in multiple myeloma as an endpoint. So two groups then, the I-Squared team and the University of Miami, really started a long-term project. Initially started as early as 2015, and I would say that the work is probably still ongoing, but there was definitely sort of a culmination in 2024. But these two groups put together and acquired patient-level data to really evaluate the association of MRD, minimal residual disease, with long-term outcomes of progression-free survival and overall survival in patients with multiple myeloma. Ultimately, they were able to collect data from over 11,000 patients and submit this to the FDA for evaluation and assessment as to whether or not it would be appropriate to use MRD as an endpoint to support regulatory decision making. And I really want to just highlight here that this was an example of a lot of collaborative efforts across multiple stakeholders, but this was a really heavy lift, and really deserves a lot of congratulations, but also sort of recognition of how difficult or challenging this can be. And so just to provide a little bit more detail as to what the actual analyses included, again, they had individual patient level data that was submitted, and they looked at both the individual level associations and trial level associations. And so when we're talking about the individual level association, we're looking at on the individual patient level, the association between MRD and long-term clinical outcomes. So is this indicative of, is MRD negative CR prognostic for progression-free survival, overall survival on an individual patient level? And then trial level associations, when you're combining multiple trials, looking at the strength of the association between the treatment effect on the trial level of MRD and on the true endpoints of progression-free survival. If we improve treatment, if the treatment improves MRD negative CR, would a similar improvement be observed in progression-free survival or overall survival? And so if there is demonstration of strong trial level association, that is deemed to be considered supportive of a validated surrogate endpoint, which then could be used to support traditional approval. Specifically in the oncology space, there's very few examples of this. And most endpoints that are used in oncology have not really been shown to have trial level surrogacy. And in fact, will have very weak trial level associations often. So this is sort of the gold standard, but it's one that's rarely seen in oncology. So specifically for the meta-analysis that was submitted by the two groups, they had enough data on essentially 11,000 patients and allowed for 25 comparisons, and in different categories of patient populations: newly diagnosed transplant eligible, newly diagnosed transplant ineligible, and then also the relapsed refractory patient population. And when looking at the results, it was demonstrated that on the individual patient level, there was very strong association between MRD and progression-free survival and overall survival, really reinforcing that minimal residual disease and multiple myeloma is a strong prognostic factor for an individual patient. When looking at the trial level associations that were evaluated, in the newly diagnosed transplant ineligible, there was moderate association that was observed between MRD and progression-free survival. There was no association really observed for relapse refractory, and there was weak to moderate associations observed for overall survival. So this was presented at an advisory committee meeting in April 2024, and the committee was asked specifically, does the evidence support the use of MRD as an accelerated approval endpoint to be used in multiple myeloma clinical trials? And the committee voted resoundingly 12-0 in favor or support of the use of MRD to support accelerated approval. And some of the comments that were shared during the committee meeting itself was that while the individual level associations were strong and support MRD as prognostic biomarker, the trial levels, as we saw, were weak or moderate. But if trial level surrogacy had been demonstrated, that this would be supportive of MRD as a fully validated surrogate, and that the standard really isn't needed for accelerated approval. And this quote from Dr. Anderson was just that it was a great day for myeloma. Subsequently, the FDA published draft guidance on minimal residual disease in multiple myeloma, and this really sort of outlined that MRD could be used as an endpoint to support accelerated approval. It went over various considerations for trial design, statistical considerations, assay considerations. And then additionally, there was a discussion of CR that could be used as an endpoint to support accelerated approval, given that in the data that was ultimately submitted to the agency, there was enough information there regarding both MRD and CR complete response that could be assessed to use either really as an endpoint to support accelerated approval. So that was published recently in support of this. So in summary of the how, of how of MRD. There was broad community stakeholder interest in development of MRD as an endpoint to really expedite drug development, given the unique situation that multiple myeloma is in at this stage. And then there was a lot of harmonization around data standards, identification of acceptable methodologies, large-scale partnerships, really, between academia, industry, cooperative groups, advocacy groups to really pool the data set, and then allow for that to be interrogated to assess for its adequacy to support regulatory decision-making. And then lastly, I'll just clue that there was early and frequent engagement with regulatory authorities, that they were really involved throughout this entire process of development of minimal residual disease as an endpoint. So what's next? One of the aspects that keeps coming back and keeps coming up is that development of an endpoint or a biomarker for regulatory purposes, it shouldn't be considered a one and done, but it's really an iterative process. And I think that's sort of where MRD and multiple myeloma is. There's definitely some outstanding questions, including can MRD be used with other therapeutics or in other disease settings, such as CAR T therapy or in smoldering multiple myeloma? The meta-analysis that was conducted did not include any CAR T therapies, and so there is some data to suggest that MRD kinetics may be different with those therapies. And so the guidance specifically that was published by the FDA outlined that additional data would be needed to use MRD in the context of CAR T therapy. And so how do we move the ball forward with that? What data is needed? What would be useful to support, the use of MRD with CAR T therapies? It seems somewhat challenging to envision needing a complete other new meta-analysis. And so how do we bridge that gap from the information that we currently have to extend to other settings, including smoldering and other settings? Is there enough data now to perhaps have a more successful trial level validation? And then also assessment of other assays. These were bone marrow based assessments in the meta-analysis. And so are there opportunities to look at ctDNA or other blood-based MRD measurements? And so these are sort of the next steps that I sort of foresee for MRD and multiple myeloma. So in summary, novel endpoints have considerable potential to expedite drug development. The development, though, and validation of novel endpoints is a really heavy lift. The novel endpoints should be fit for purpose, and there should, in many instances, to justify this lift, be a real need for endpoints. And that really sort of allows the community to come together to sort of assist and move these endpoints forward. And lastly, that it's not a one and done process. It really is one that I think over time, continuing to advance the field is necessary. So that concludes my presentation. Thank you very much.
Thanks, Nicole, for your presentation. Next, we'll hear from Shraboni Sharkar, who will be presenting virtually. Shraboni is the Global Medical Affairs Lead for Neurology at Roche Diagnostic Solutions, Switzerland. Shraboni holds a doctorate in molecular biology, focusing on neuroimmune pharmacology. For the past 10 years, she has been working at Roche Diagnostics, gaining extensive expertise in the clinical implementation of high-value medical diagnostic assays. Shraboni, thanks for joining us.
Thank you very much for the invitation, and good afternoon from London. I'm actually at a very important Alzheimer's disease conference, so this is really a good time to connect and share how we are advancing diagnostics in neurology at Roche. So if we can maybe go to the next slide. So in this presentation, I'm going to talk about the Neuro Toolkit. So it's one of our tools that we use basically for research purposes. But then I want to show you how this has helped us to come up with biomarkers for clinical use now that is being used for patients. So first, I would like to give you a bit of brief overview of how we drive neurology at Roche. So we are an IVD company, so we bring out tests which are in vitro diagnostics assays, which means that they have been regulatory approved to be used for patient use in different markets. So our primary goal is to bring the best possible IVD assays to the market. However, to develop the highest quality IVD solutions, we need robust evidence generation. And we achieve this through a journey that begins with identifying a reliable biomarker. Then we progress to developing the right assay. And then ultimately, we leverage the combined power of the Neuro Toolkit, as well as assays which are used for research use only. This allows us to fulfill our promise to patients, bringing assays to the market as quickly as possible. So if you could go to the next slide. And actually the next one. So first we identify diagnostic needs, which involves extensive discussions with experts and key opinion leaders across the neurology spectrum to pinpoint unmet medical needs. So once we identify these unmet medical needs, we target the key markers that address these gaps. Afterwards, we develop the assay using selected optimal antibodies and roll it out on our Alexis platform. That's a platform that's used for clinical use, which you may be or maybe not familiar with, as it powers all of our IVD assays. So once rolled out, we use the Neuro Toolkit, or the NTK, as I will refer to from now on, to generate the necessary evidence to inform and guide our IVD development. If you can move to the next slide. If we could go to the next slide, please. Sorry. Could we go to the next slide? I don't know if there's a lag on my thanks. So switching to the next phase of the journey, which is assay development on the Alexis platform, the top biomarkers are validated on our automated platforms. This is completely automated, very little user, very little need for a user, enabling standardization, ready-to-use reagents, a wide footprint, and fast analytics. Now, if we could move to the next slide, looking at how the NTK supports this journey, we're bridging the gap between exploratory studies and clinical practice and commercialization. We do this by utilizing different labels. So the RUO, the research use only, are labels for exploratory studies, PEO or performance evaluation only. This is for clinical and registrational trials, and ultimately it's the IVD that support routine clinical practice. In the following slide, it highlights that we are focusing more closely on the Neuro Toolkit. Its success stems from three main pillars. The first pillar is our global biomarker footprint. So we have multiple testing sites operating under a single unified NTK study protocol. So we currently measure across 4 testing sites, 3 in Europe, 1 in the US, which helps us generate high quality data through true collaboration with different experts and diverse studies. And as you can see, this comprises of our portfolio in both CSF and plasma at this point. But again, this keeps changing as we add new biomarkers to our portfolio. And if we can go to the next slide, the second pillar is our widespread partnership network. So the true strength of the NTK lies in how we collaborate across pharmaceutical companies, philanthropic organizations, and academia. And the final pillar, alongside the biomarkers and collaborations, is the data itself. All the data generated must be analyzed with the right tools, and this is where the NTK app comes in, featuring dedicated curation, analysis, and meta-analysis applications. This helps us integrate, analyze, and aggregate data to make effective comparisons across different cohorts. And then to conclude this journey and fulfill our promise to patients, we're bridging the diagnostic solution to improve patient journey. So what we have just outlined is our solution for evidence generation to create the best possible IVD assays. And beyond that, we also want to highlight the real-world impact of integrating the NTK. So we would like to share some concrete examples of biomarkers that through the NTK have successfully transitioned through into IVD solutions, directly supporting clinical practice and making a tangible impact on patients today. So if we can move to the next slide. So today, I think if we can go to the next slide, please. I don't know if there's a lag on my end, but so today I'm going to talk to you about our Alzheimer's disease portfolio, and particularly one assay in particular, which is the p-tau217. So what you see right now is our current on-market Alzheimer's disease portfolio. And most of the tests that you see here have been developed after evaluation and evidence generation through the Neuro Toolkit as well as parallel studies. So as an example, I'll talk about the p-tau217. So if we could go to the next slide. So p-tau217 is the most recent and the most awaited addition to our portfolio to complete the current diagnostic pathway needs in Alzheimer's disease. And it has received the CE regulatory mark just a month and a half ago, so early May. It is designed to deliver high accuracy, operational simplicity across care settings, meaning both in primary and secondary care settings, and enhance clinical clarity for early stage diagnosis. So if you look at the image on the right, looking at the data from the Global Biomarker Consortium, a round-robin study, Alexis p-tau217 emerged as a top-performing plasma assay for detecting amyloid pathology. So if we can go to the next slide. Yeah, so now let's look at what is this particular test and how can it be used. So these are the specific CE cutoff values established for the Alexis p-tau217 plasma assay. As you can see in the graphic, the assay has a dual cutoff. So it means that it categorizes patients into three distinct groups: a high cutoff above the higher level, which is 0.38 picograms per milliliter, indicates a positive result, meaning a high likelihood of amyloid pathology. A low cutoff or below the 0.223 picograms per mil indicates a negative result, showing a low likelihood of pathology. And values falling in between these 2 points are considered the intermediate zone, where further testing is recommended to clarify the diagnosis. And generally, this is done with the current standard of care, which is CSF via lumbar puncture or PET, amyloid PET analysis. So if we can go to the next slide. Yeah, actually, I'll come back to this later, so we can maybe progress to the next slide, because I would like to give you a little bit more details on how this cutoff was determined and validated. So some details on the study population. So the data presented is derived from our clinical performance study, which evaluated a robust sample size of almost 1,000 participants across two multi-center cohorts, namely the approach cohort, which was a low prevalence cohort, and the AACI cohort, which is a high prevalence cohort. And combining these cohort gives us a diverse range of AD prevalence. The average age of recruitment was around 71 years. The study enrolled patients presenting with cognitive complaints or objective memory impairment of unknown etiology. And to ensure that the data reflects routine clinical practice, participants with potential comorbidities were not excluded, of course, provided that they did not present with active symptoms such as delirium or encephalopathy. So the charts below, as you can see, they break down the characteristics of the study population. So the first graph shows that approximately 40% of the cohort was amyloid PET positive, while 60% was negative. Gender distribution was relatively well balanced, 55% female, 45% male participants. The study spanned both primary and secondary care, with most of the patients being managed in secondary or tertiary care as expected, with the 75% and the remaining 25% evaluated in primary care. In terms of memory status, was tested with the mini mental state examination. The scores show a broad range of baseline cognitive functions, so with the largest segment falling into the mild to moderate impairment ranges. And the study was also powered for early dementia and MCI. So now if we can go to the next slide. So here I'd like to highlight the clinical performance of the p-tau217 plasma assay. As outlined in the table, the assay demonstrates robust clinical utility across care settings. So both, as you can see, both sensitivity, specificity consistently remain above 93%, regardless of whether it's in primary care or secondary care. So for instance, in primary care, which is approximately at a prevalence of 20%, the established cutoff achieves a very high sensitivity and a very high negative predictive value, like more than 98%. While in secondary care, at a prevalence of 45%, the performance remains highly robust with high sensitivity and a very, very high PPV, positive predictive value. So for both settings, the intermediate zone falls within the 20% range. Then furthermore, as you see in the criteria on the right, the assay successfully meets the rigorous CEOI consensus recommendations for acceptable biomarker performance at both 20% and 50% prevalence of Alzheimer's disease. This confirms its reliability as a high-performing diagnostic tool for evaluating patients with cognitive impairment. So if you go to the next slide, which I believe is our final slide. So what I would like to highlight here is how this particular test that started with the Neuro Toolkit now has regulatory approval, probably soon also in the United States, is now available for clinical use. And the vision is this for the future patient pathway is to streamline and thereby enable faster diagnosis, because one of the biggest unmet needs in Alzheimer's disease is time to diagnosis. And we believe that an easy to use plasma test can help with that. And this is a pretty simplified illustration of the potential pathway, but we believe that it could potentially look like this in a few years. So at primary care during neuropsychological testing and other blood tests, beta 217 can be used to rule out Alzheimer's disease with the high negative predictive value that you just saw at a low prevalence setting. And patients who are not ruled out can be referred to a specialist. So there, if they fall in the indeterminate zone, they can be reconfirmed by CSF or PET. And patients who are ruled in could be either confirmed by CSF or PET, or the blood test can be considered confirmatory. As you saw, we do comply with the CEOI criteria. But again, this depends on the clinical practice recommendations of the particular country. And then these patients can then be evaluated further for anti-amyloid therapy. So to conclude, research through tools such as the NTK has provided us a faster and evidence-based path to bring valuable solutions to our clinicians and patients. So I hope this example has shown you the value of doing research with such tools. Thank you.
Thanks, Nicole and Shabroni, for those presentations. We will keep these biomarker experiences in mind as we move into session 1, where we will be discussing approaches to operationalize fit-for-purpose biomarker evidence across regulatory uses. I would now like to invite my colleague, Rachel Hendricks-Stirrup, to the stage to introduce and moderate our next session. Rachel.
All right, welcome. Thank you all for being here. Uh-oh, I've got 2 microphones. I think I'm using this one now. Okay, great. Awesome. Again, thank you all for being here. We are going to move into session 1 now that we have opened up with our intros and use cases. As I've been introduced, my name is Rachel Hendricks-Stirrup. I'm the Research Director of Real World Evidence at the Duke-Margolis Institute for Health Policy. So for this session, we are going to focus on the preliminary policy roadmap, looking specifically at Goal 1. We're going to look at, within the roadmap, under Goal 1, recommendations to support biomarker evidence maturation as biomarkers move from exploratory or supportive uses towards more consequential regulatory roles, as we've highlighted a bit today, including use as surrogate endpoints in individual product submissions. So as we begin, we're first going to open up with a presentation We have Pujita Vaidya. Please correct me if I pronounced that wrong. Who will highlight the modular acceptability framework for biomarker development. So Pujita serves as Regulatory Science and Policy Director at Sanofi. She leads efforts to advance policy topics, including novel endpoints, innovative approaches, AI or artificial intelligence, model-informed drug development, and PFDD implementation. In her role, she also partners with cross-functional teams to embed PFDD and other policy principles across development programs, and collaborates with regulators and external stakeholders, which we heard is critically important today, to advance policy priorities around biomarker development. So I'm going to welcome Pujita to the stage, who's here with us in person. Thank you for being here, and I'm going to turn it over to you.
Hi, everyone. First of all, thank you to Duke for inviting me to present on some of the interesting work that we've been doing when it comes to advancing novel endpoints. So as Rochelle mentioned, I'm Pujita Vaidya, and today I'll be talking about this work that we've been doing and show a conceptual illustration of an acceptability framework proposal for all of you to ponder on. And this is work that I'm presenting on behalf of a small regulatory policy working group that came together, which includes Biogen, BioMarin, BridgeBio, three bios, just came together to really think through, what the problem is and how can we move forward. So to start off with, despite decades of novel endpoints, including biomarker science that's been developing and we've been working on, there still continues to be this sort of persistent translational gap that exists between endpoint discovery and regulatory acceptance, which mainly is driven by inconsistent maybe methodologies or unclear evidentiary standards. And when we're looking at it, the three key areas of misalignment that we identified are biological and physiological relevance, so the mechanistic plausibility and consistently demonstrated across submissions. So this is creating this unpredictable regulatory outcomes. The next is clinical utility, which is more along the lines of, mechanistic plausibility really expectations for validation of novel endpoints that matter most to patients overall. And then finally, evidentiary robustness. So that evidence often fails to demonstrate novel endpoint, let's say fitness for intended regulatory context of use. So what is the opportunity? The opportunity that we identified is this harmonized approach that's necessary so that the field doesn't remain trapped in this cycle of inefficiency where scientific advances stall at a translational stage, development timelines lengthen, and patients, particularly those with rare or complex diseases and high unmet need, they don't need to wait longer for precision diagnostics and therapies that already have, the science already exists. And I think we're here together, FDA, industry, academia, communities, patients, the stakeholder community, we need to work together to advance this. For the purposes of my presentation, I'll be referring to novel endpoints, and I just want to note that that does include biomarkers when I speak to it, and I won't go into all of that. So what we need to remember is we're not starting from scratch here, right? FDA, industry, and the broader ecosystem, we have already built some of these pieces that can be worked towards a solution. We have FDA frameworks, whether it's the best taxonomy, the biomarker qualification evidentiary frameworks that are there, the fit for purpose validation principles that exist. We have cross-stakeholder learnings. There's a lot of public-private partnership work that's been done. Insights from patient-focused drug development, clinical outcomes work that's happened, the FDA's guidances. We have precedent examples as well. These emerging use cases where endpoints have progressed across disease or population or context of use. So really being able to offer that practical evidence patterns for us to build from. And then finally, decision science principles. FDA's prior use of these structured risk-based evaluation to focus evidence generation on what matters most for regulatory decision making. And examples here are the quality by design work, the benefit risk work, framework that FDA has, all really rounded on decision science principles overall. So we still have a challenge though. And what we did as a small group was think through and map out, these challenges that we're seeing in the ecosystem, and finally identified these into three main issue clusters. First is around biomarker endpoint development and the challenges there. So scientific and technical uncertainty, process structural and incentive barriers, and a lack of a scalable model overall. The second is around engagement and communication gaps. So are some of the pathways that we have maybe more IND focused only and drug specific, or sometimes shifting views and inconsistency and some of the advice that we get overall. So really thinking about that. And then third is around DDT program limitations, which includes the biomarker qualification program, somewhat of a narrow scope, undefined timelines, the qualification and IND gap. So when we think about all of this, what I want to focus on now as we move forward is that where we believe is the greatest impact that we can make is really focusing on the biomarker endpoint development challenge on the far left here. And specifically the scientific and structural uncertainty around what evidence actually is needed. And we picked this because this is foundational, right? If sponsors don't know what evidence needs to be generated, then better engagement pathways, DDT qualifications, like any of that overall reform may not eventually matter. So we really need to start from that foundation. And we were also aware of other ongoing efforts let's say in the qualification space by Friends of Cancer Research, you'll probably hear about later. So we wanted to think through what can be a companion solution to what's ongoing. So the proposal. We propose a structured framework, acceptability framework, that can tell sponsors and regulators exactly what evidence is needed for what type of endpoint in what regulatory context. And the purpose really is to focus our resources, regulators and sponsors' resources, on generating the most critical evidence that's required to establish novel endpoints across diverse regulatory scenarios by creating clear linkages between scientific objectives that we have and the regulatory requirements that are there. And the key question that we're trying to solve for is how do we systematically bridge that gap between the evidence we have now, today, and the evidence that regulators will need in the future. And the main objective is to develop a structured framework that categorizes evidentiary requirements by endpoint type and regulatory pathway, identifies specific data gaps so sponsors know exactly where to invest and where perhaps we need to refocus, define validation needs relative to the endpoint's intended context of use. And what I'll do is walk through some of the core concepts and the structure overall. We started by grouping the common purposes and the scenarios we see across drug development into what we call endpoint archetypes. So there are two dimensions to this, the type of endpoint and how its use might shift over time. First dimension is the endpoint type, right? Here we've identified 6 categories that determine sort of that baseline expectation. Clinical endpoint, surrogate, intermediate, endpoint based on DHT captured data, so that we have the endpoint type there. And we're really thinking about this holistically, not specifically focused on, surrogate endpoints or whatnot, but really thinking about it holistically. Then we have the endpoint modification, and that's the end type and endpoints type may stay the same, but its use can shift overall. And the 4 scenarios where that endpoints that we identified, where the endpoints tend to shift, is around, let's say, first, cross-disease translation. So endpoint moves from one disease to another with the similar pathophysiology. The context of use, expansion. So endpoint moves from one regulatory context to another So enrichment to primary endpoint, let's say. Disease stage translation, endpoint moves from late stage to early pre-symptomatic diseases, and a clinical measure adaptation. So established measures is modified for use in trial endpoints to better capture treatment effect. And where the uncertainty may not be about, whether an endpoint is novel or not, but it's more about, so on which modification scenario applies and what evidence that is required overall. Now, the other next core concept I want to think about is how much evidence is actually needed, right? And it can depend on the regulatory precedent. And the precedent can reduce the burden that's there. So the more regulators and sponsor see an endpoint work in similar contexts, the less the new evidence perhaps may be required. And here, just looking in the middle is what we're thinking about, the different precedents. If it's never been used, there's no regulatory precedent, there may be a need for more of a full validation evidence package. Significantly different context of use if it's a similar context of use. So really thinking through that, and then thinking about the amount of data that's necessary for that. So what this gets us thinking about is how do we right size that evidence to the regulatory decision context and not necessarily to the novelty of the endpoint itself. Then let's look at the level of evidence. Then necessarily, if you think about this, the level of evidence should then increase with the risk that's there. And as we thought about this, we broke it down into these modular elements. So very much aligned with, let's say, FDA's totality of evidence approach to focus on the most critical elements of regulatory uncertainty for a sponsor to address. So each module here can be considered a distinct risk element. And we need to identify where that uncertainty lives and focus our resources there overall. So the 5 modular elements is first the analytical and clinical validation, which we've grouped together. I have grouped together in this pyramid to show that, this is really the foundational element in this, and it's fixed and it's required. And what this gets us is, does the measurement tool work? And does it reflect meaningful biology? Then we can think about regulatory use. What regulatory decision does it drive? Is it, primary endpoint versus dose selection? Then context of use specification. When, where, in whom, for what purpose is the endpoint being applied? And then the measurement adaptation. And if the tool is modified, does it still mean what it used to? And then the same module can be, one specific module can be high risk or low risk, depending on the context overall. So an example I want to give here is, let's say if we take regulatory risk, there may be higher risk when the endpoint drives the major decision, let's say serving as a primary endpoint for approval, and lower risk potentially when the endpoint supports a lower stakes decision. For example, informing dose selection rather than the ultimate benefit risk determination. So what this can help do is, when sponsors and FDA can look at these 5 modules together and agree, this is where we're confident, this is where we're not, this is where we need to really focus on this evidence generation, or, to really get that overall picture. And when I talk about the overall risk picture, what we did was we were thinking about, we've established this risk to regulatory acceptance can be modular, and then we can then map this out. So when we map out the risk modules against the endpoint archetypes we've defined earlier, what we can do is then perhaps predict evidentiary needs. And so here you can see an illustration of a risk weighting matrix. So along the vertical axis, you have the endpoint archetypes. And then along the top, you'll see the 5 risk modules. And this is really meant to be a practical tool to help get us together to think about what's necessary in a certain modification scenario. And then we go on to apply these risks. So here you'll see we have high, medium, and low. And what we're assigning this risk by is being two main risk factors that we took into consideration. There are many more out there. Leveraging prior work. So how much existing evidence can be carried forward? How has the endpoint been used in a similar context before? And then what is that uncertainty there? What areas of uncertainty are there? What new questions does this specific archetype raise? And then what gaps remain that could impact regulatory confidence? So the thinking is that by starting with these considering these two factors, then we assign these various high, medium, low risks. And I'll walk through an illustration here on what I mean. And it's using the cross-disease translation as our worked up example. This is a scenario, just to remind folks, is where we have the endpoint validated one disease and is applied to another with a particular similar pathophysiology. And what I want to show here is like for each module, we can ask the question, what can we carry forward and what genuinely needs new evidence? So when we look at the analytical validity, what can we leverage in this worked up example? We have, existing assay validation. Let's say that exists, and the measurement tool itself has already been characterized. But there is a lot of uncertainty. Fit for purpose, is it fit for purpose in the new disease biology, cross-population variability, drift and bridging? So we're seeing a lot of uncertainty, let's say. For that reason, we assign it a high, so it's a red. Then we move on to clinical validity, similar types of considerations. What can we leverage and what's uncertainties out there? So for each one of these, sort of walking through and assigning specific risks to that. And then from there, this gives us a picture, right? So if you look at this overall for cross-disease translation, the measurement tool is not, it's helping you understand. Maybe we should really focus on analytical, clinical, and context of use. Those are the areas where it's high, and we need to focus on that. So it's really trying to give that overall big picture to understand where do we need to invest, where do we need to build or and be used as a conversational tool, a communication tool between sponsors and regulators, internal decision making, and overall. And then, so with that, you sort of saw the thinking, the concepts, the framework, and action, and it is really what we see this as a starting point, right? Not a finished product, and and that's the reason why I'm here representing our small group is we got together a small group to think about this, but we want to see whether it resonates. And we need a larger group of stakeholders to actually look at this, pick it apart, but work through and think through the thinking. But we wanted to demonstrate that this is one way for us to look at it. If it resonates, let's try to build this out together. And it is really meant to be designed to provide greater regulatory predictability and efficient development, support both sponsors and FDA efficiency, provide expectation guardrails, and encourage leveraging prior work to have it be more of a scalable model overall. So with that, I will pause there. Thank you.
Hey, I think we have to do some reconfiguration really quickly. As we do that, I'm going to welcome our panelists to the stage. So we're going to move into the discussion portion of this session for today, and we're going to discuss a couple of things highlighted in today's agenda. So we're going to discuss how evidentiary expectations should vary based on a biomarker's intended regulatory use, degree of novelty, context of use shift, assay or measurement adaptation, and residual uncertainty about clinical relevance. And we're also going to discuss additional expert and as well as your input today within our audience, both online and in person, on practical frameworks, evidence development archetypes, as we learned about today, just now in Pujita's brilliant presentation. Thank you again for that. And implementation challenges, particularly in rare disease or accelerated approval contexts, should that extend into other areas. Happy to discuss that as well with everyone. So now that we're all seated comfortably, I hope, I'm going to introduce everyone. So first, we have with us, sorry, 1 second. I want to make sure, I think we're sitting in a different order. So I'm just going to introduce everyone by name, and you can just raise your hand as I say your name so everyone knows who you are, both online and in person. So we have Amanda Klein, who's the founder and principal consultant at Biomarker Solutions, LLC. Next, we have Issam Zineh, who's the director of the Office of Clinical Pharmacology at the US FDA, or Food and Drug Administration. Finally, we have Jad Zogby, who's the head of Biomarker Analytical Sciences at Biogen. Thank you all for being here. I'm really excited about this panel. As the director of real world evidence, I love talking about that. And as a pharmacologist, I love biomarkers and talking about that too. So each of you are going to share some opening remarks as we begin our discussion, starting with Amanda. So Amanda, I'm going to turn it to you. And then next, we'll pass it to Issam.
Great. Well, thank you again, Duke, for having me participate. This is, of course, very meaningful for all of us. And my always spiel is biomarkers are some of the misunderstood terminology, but we use biomarkers in everyday life. When we go to the doctor, right, we get a lab test, check for cholesterol to see our risk for heart disease. That's a biomarker. If we start a new drug and we need a urine drug screen, that's a biomarker for, illicit substances. So the things that we can use biomarkers for can be on a population health level, but also for ourselves, for our own individual care. So with this first session, one of the key parts that we're talking about is that idea of how to think about that biomarker evidence generation and how it can evolve over time. And one of the important components that I see as a consultant is a lot of companies want a surrogate endpoint. We want a surrogate endpoint for our drug. We want reasonably likely, we want accelerated approval. And so fundamentally, regardless if you have a reasonably likely surrogate or a fully validated, you need to link biological plausibility to a clinically meaningful outcome. Okay? And so that is fundamental. So when I think about the beginning and I think about a lot of natural history studies, that is the prime time to really engage diverse stakeholders in natural history planning studies, prospective data collection that can then be used to inform future for that surrogate endpoint development. So we'll talk more. The other part I was going to just really echo is the 2 case examples this morning. It's so powerful because there is so much lessons learned that regardless if you're in a different therapeutic area, you can really take that information and then apply it and think through for your own program. So one thing we'll talk about in this and throughout is the importance of knowledge sharing, the good and the bad. And that way we're not reinventing wheels, but we can learn from both successes and failures. So thank you.
Yeah, thank you, Amanda. Next, Issam.
Yeah, thanks very much for organizing this. What I thought I'd do is maybe just provide my own sort of reactions to the opportunities list, which I understand will hopefully become a roadmap. Places where I agree and places where I maybe would push back a little bit, and hopefully that'll be, certainly relevant to our session, I hope, but then maybe some substrate for later today as well. A laudable diagnostic effort. I think that the document does a really nice job sort of laying out some of the problems with biomarker development and implementation, and certainly a sensible scope. I think circumscribing this to biomarkers that are to be used in sort of individual development programs and submissions makes a lot of sense. I do think, and this is sort of the perennial problem with discussions around biomarkers for regulatory use, that we often risk treating the central challenge as one of a regulatory problem or a regulatory transparency. I think Nicole's example earlier today shows that there's a bigger problem, and that is biomarker development is hard. the drug development ecosystem and even the translational biomedical research ecosystem is not set up for multi-interventional longitudinal studies linked up to outcomes that get you where you need to be in terms of surrogate endpoint, which seems to be the sort of the unicorn that everyone is chasing. So, that's very much I think an important reality to keep in mind as we're trying to solve problems related to developing a robust evidence base. I think this roadmap correctly treats biomarker evidence as a continuum. I love this idea of residual risk. I think it's very much an important concept, and evidentiary standards or requirements should really be driven by tolerance for residual uncertainty calibrated to the question that the biomarker is intending to answer. I don't personally love these categories of exploratory, versus supportive. I don't even know what those terms really mean. And I also even think reasonably likely surrogate endpoints, that the evidentiary expectations could be different depending on what the clinical development context is. I do think it also is wise to separate measurement validity from clinical validity, although we can talk a little bit more about sort of what the problem is and what the intersection is between those. I also think the roadmap handles this idea of regulatory inconsistency carefully. Oftentimes we hear this thing about regulatory inconsistency, or what appears to be regulatory inconsistency, and that's driven by inconsistent outcomes. There may be, sort of, why was this biomarker accepted but not this one? So on face, it might look inconsistent, but what we're really driving for is consistency in the thought process. The outcomes may be different, and they need to be appropriately contextualized, but it's the consistency in sort of the evidence assessment. I think that's sort of a more appropriate goal. Some of the transparency recommendations in the document are, I think, practical and have some potential high value. And this exercise does understand that the problem is partly infrastructural, and a big part of it is infrastructural, I would say, not merely regulatory. Where I would push back a little bit, and maybe this will be fun for discussion, maybe this is predictable, but a lot of the action items and the burden still falls on FDA, and with the caveat that, I know we need to prioritize this. There is still a huge inventory that is a set of FDA-directed tasks. Even though if those were squarely within our remit, which I would argue they're not, they're really community propositions, it does underestimate the kind of the operational costs of offloading this effort on FDA. And then the feasibility of some of these asks, given that we have to maintain, confidentiality within development programs, that needs to be considered. But I think more importantly, that sort of over-reliance on, well, this is a regulatory issue, it obscures where many of the bottlenecks are that are independent of FDA. And this has to do with the scientific maturity of the biomarker, internal sort of sponsor fragmentation. There's as much within company and across company inconsistency in approaches to biomarkers as there is, as maybe even more so than there is on the regulatory side. Late assay planning, reluctance to invest in a particular biomarker before having regulatory certainty on acceptance. So there's this chicken and an egg problem as well. So I would think that that's something to look at. Obviously the central evidentiary framework needs to be worked out. So I really applaud Pujita and the working group for putting something out there. And then just one or two other quick things. This idea of earlier engagement and centralization of models for evidence evaluation. We just need to think about whether or not those are redundant with already existing modes of engagement with regulators. And finally, I think at least nominally, the roadmap tries to separate this idea of biomarkers development within a development program. And it sort of separates that from biomarker qualification kind of writ large. But then as you start to get to later recommendations, that boundary becomes a bit more porous. So I think we do as a community need to be thinking about kind of where do we want to put our efforts in an evidentiary framework and the infrastructure that it takes to get there, or in sort of more discussions around formal qualification programs. So I'll leave it at that. Thanks.
Awesome. Thank you so much. That was very insightful, thought-provoking, I think, for many of us here. I'll turn it to our final panelist for opening remarks. Jack.
Thank you, Rachel. Thanks, Duke, for inviting me to be part of the panel. So I'm here with Biogen, but I'm also representing biomarker leaders within the APS Translational Clinical Science Working Group community with APS. And this is pretty much how we got engaged. We got engaged because of the importance of the analytical validation, the analytical aspect in everything that we do when it comes to generating the right evidence. Evidence is, you heard Sam mention consistency, inconsistency. So the data needs to be consistent. The data needs to be sensitive, needs to be specific, needs to be precise, because that's essentially what makes our work defendable and creates that evidence long term. So the importance of analytical validation is pretty much what I'll be discussing as part of this panel. I'll focus more on the data generation and how the rigor that it needs to really define be part of the roadmap and define the evidence needed for surrogate endpoints or reasonably likely surrogate endpoints. That's it. I'll keep it simple. Thank you.
Okay. Awesome. Well, thank you to each of you for your opening remarks. Pujita, thank you for the opening presentation for this session. So now we're going to move into our discussion. So I'd like to call everyone's attention to our Slido poll that I believe will pop up on the screen shortly. There we are. Okay, if you can't access the Slido poll directly through the QR code by scanning it, particularly for those of you here in the room or joining us virtually. You can join the poll at slido.com, #bio, B-I-O, B as in boy, I-O. And we're going to leave this slide up for a moment to allow everyone a chance to enter the poll and complete it. But essentially, the poll asks, among the list of recommendations provided, please select 3 to 5 that you feel would be worth discussing further or considering further for today's discussion. So I can go over some of the options for everyone verbally, if that's helpful. First option is develop a regulatory framework that operationalizes evidence staging, reflects how biomarker evidence develops, and integrates risk-based evidentiary expectations, iterative validation, and biomarker lifestyle management. For this goal, the second proposed recommendation is distinguish evidence that is decision critical from evidence that is supportive, exploratory, or context dependent, as biomarkers are used to support progressively more consequential regulatory decisions. Number 3, clarify when surrogate endpoints may provide informative support evidence, even when they are not sufficient to serve as the primary basis for approval. Number 4, clarify how analytical validation of a biomarker measurement tool can be evaluated separately from clinical validation of biomarkers' intended regulatory use. Number 5, describe how sponsors can build evidence packages that integrate disease biology and mechanistic rationale, natural history evidence, longitudinal biomarker trajectories, assay performance data, available clinical outcome data, and appropriate comparators. I know that's probably a mouthful, but it's all important. Number 6, clarify how evidentiary expectations should be adapted when rare disease programs cannot generate separate biomarker validation studies and treatment effect trials. Number 7, clarify how sponsors should define a biomarker's context of use at an appropriate level of specificity so evidentiary expectations remain decision relevant without fragmenting biomarker development across overly narrow use cases. Next, clarify how reasonably likely surrogate endpoint expectations differ from full validation expectations in rare disease accelerated approval contexts. Number 9, clarify evidentiary expectations for biomarkers used in pre-symptomatic or asymptomatic populations where clinical outcomes may be delayed or difficult to observe. Number 10, identify common biomarker and surrogate endpoint development archetypes like cross-disease or disease stage translation, context of use expansion, and assay adaptation, and describe what prior evidence may be leveraged and what residual uncertainties remain. We have plenty here, but I want to make sure I verbalize all of them again to accommodate everyone in the room and online. Provide guidance on appropriate analytical validation to ensure generated evidence supports a biomarker's context of use. Number 12, clarify evidentiary bridging expectations for biomarker data generated using non-identical assays, platforms, tracers, or technologies over time. Number 13, prioritize early development and characterization of the biomarker assay or measurement method, including comparability, bioanalytical validation, and cross-team continuity when a biomarker may later support a more consequential regulatory role. 14, prospectively design natural history studies or biomarker collection plans to support future quantitative interpretation of biomarker change. Number 15, report evidentiary burdens that commonly limit biomarker use, including biological plausibility, the biomarker change, and clinical benefit relationship, clinical relevance, and longitudinal biomarker evidence, analytical validation and assay comparability. And number 16, clarify how predefined evidence development and bridging plans can support anticipated changes in a biomarker's context of use, assay, measurement tool, or analytical approach over time. So we'll give everyone just a moment as I've read through each of those to respond. It looks like we have 58 responses so far. Okay. All right, we have results. So out of all of the 16 recommendations that I described or verbalized for you all, we asked you to select 3 to 5 that warrant, that you feel, in fact, warrant further discussion or consideration. And it looks like we have a top candidate here. So describe how sponsors can build evidence packages that integrate disease biology and mechanistic rationale, natural history evidence, longitudinal biomarker trajectories, assay performance data, available clinical outcome data, and appropriate comparators. So why don't we start there? Considering that that is the top rated discussion point, I want to give our panelists a chance to react to that. Now, we've talked a bit about this already. From each of you have commented to some more or less extent on this, but looking at this particular selection as our top rated, followed by developing it looks like, okay, yes, develop a regulatory framework that operationalizes evidence staging, reflects how biomarker evidence develops, et cetera. Looking at these two as our sort of top rated areas, what are your immediate reactions or thoughts to this based on 83 responses or votes from the audience? Whoever would like to react first.
Well, I'll jump in.
Sure.
These were my top 2 as well, in inverted order, but it's not surprising. I think these are 2 sides of the same coin. I think an evidentiary framework, number 1 here, basically serves as the foundation for the conversation. And then basically once you have a consensus on what are the things that need to be addressed, ranging from, as Amanda said, I think on the one end of the continuum, biological plausibility, which is totally fraught, as a concept, right? But then all the way through to sort of clinical outcomes, which again, we saw from Nicole's presentation, is very hard to establish in terms of a biomarker endpoint relationship for full surrogacy. But that's the continuum of the evidence. And so once you have a framework that is explicit about how those things could be articulated, then 5 becomes sort of a natural extension of that, which is sponsors then put together their evidence packages, either in terms of summarizing the evidence in support of, or developing a sort of evidentiary plan, right, to fill in the knowledge gaps. So I think these, to me, seem very complementary.
Thank you. Any other thoughts? Would you
Yeah,
go right ahead?
I can chime in. I completely agree. I have my notes here, and actually, I have these too. And also, I'm a little biased, right, with the regulatory framework. That's one of the thought that we were trying to put out there, right? A proposal for little aspects of it, pieces of it that can fit into this larger. I mean, we use the term framework, but someone recently told me like that can mean so many different things to different people. I think we're really thinking about an approach, right? An approach of how to get all of the pieces together and get it right. And as Isam mentioned, we need to get the evidence generation right. We need to figure out what's necessary and really think about that regulatory approach or framework. And then once we have that right, then we can figure out what the evidence package needs to look like at the end, right? And where I'm selfish, where to say, yeah, I think we've at least planted a seed on some of the approach and the thinking, and some of the other, other recommendations that you have, I think fit into this, right? Because there was one around distinguished evidence that is decision critical. It fits into this real framework thinking. There's another around FDA and stakeholders should identify common biomarker, like archetypes, which is some of the work that we've done. All of it sort of fits into this overarching recommendation, these 2 recommendations here. So I think this would be a great start. The question is, who's going to go out and do it?
Right. That's always the
I
question.
think that's the thing. And as Isam has mentioned, it's not just FDA, it's not just sponsors. We have to do this as a community together.
Mm-hmm. Now, you both mentioned interesting things. Isam, you mentioned that recommendation 5 and 1 here being the top 2 rated. They really are, perhaps 2 sides of the same coin. And then Pujita, you mentioned that at least one of the other recommendations might even fit into, or stage itself maybe within that coin somewhere. Whether it's in one particular pore or another pore between both sides. I don't know how to illustrate it appropriately, but I'm hearing that you're probably saying that some of the other recommendations might fit into this coin somehow as part of getting from one side of the coin to the other.
Exactly.
Do you all have any thoughts? Any others who haven't commented yet? Or Pujita, Isam, even if you have further ideas around that idea? What are your thoughts? I know we went over a lot of recommendations, and one of the reasons why I wanted to verbalize each of them is because, we also think maybe these fit all together somehow. Maybe they don't, but maybe we can talk a little bit more about that. And actually, we have a third most rated recommendation here, which is clarify how evidentiary expectations should be adapted when rare disease programs cannot generate separate biomarker validation studies and treatment effect trials. Followed by clarify how reasonably likely surrogate endpoints expectations differ from full validation expectations in rare disease accelerated approval contexts. We asked for 3 to 5 recommendations that warrant further discussion, so I'll mention the 5th, which is identifying common biomarker and surrogate endpoint development archetypes, and describe what prior evidence may be leveraged and what residual uncertainties remain. So again, thinking about the 2 sides of the same coin and where other recommendations might fit into that coin or be a part of it. What are your thoughts based on what we see as the most uprated 3 to 5 recommendations here?
I hate to filibuster, but I think many of these were related. So, for example, the rare disease opportunity, and even the one that flags how do you develop biomarkers for basically prevention, pre-symptomatic disease. I mean, those are basically just variations on the theme of context of use. If you've developed a framework, right, that speaks to the evidentiary, that speaks to what is the biomarker intended to address and in what context? You can develop a universal framework for that. A good evidentiary framework is a communication tool. We've found that in our experience. It's a near impossible task, I would say, to develop an evidentiary framework where you can plug in and say, rare disease, this prevalence, this sort of mechanism of pathology goes into this square. Common chronic disease is going to go into that evidentiary square. It's not going to be that specific. Instead, it will be, what are the concepts that are relevant across, irrespective of whether it's rare, common, chronic? What are the evidentiary concepts? What is the risk in sort of making the wrong decision, in that clinical context? What is the likelihood of and how does this biomarker, if we're focusing on biomarkers, what role does the biomarker play in the totality of evidence? Are we solely relying on the biomarker to make the decision, or is there a constellation of lines of evidence that will de-risk that decision based on the biomarker? To my mind, all of those conversations happen irrespective of whether you're talking about Alzheimer's disease or mild cognitive impairment or some rare, inborn error of metabolism. It's the same sort of schematic or framework for the conversation.
Yeah, and acknowledging the reality that a lot can be true at once, with the biomarker being one of those truths and other endpoints being part of the broader truth around the disease scenario or the clinical scenario. I think that resonates a lot and should definitely fit into the framework as a sort of conversation starter or component. Any other thoughts from our panelists? Isam, thank you for your filibuster, and we love hearing from you, but I want to make sure that, we give everyone here a chance to comment on what we're seeing here, what we're hearing, and where the conversation should go from here.
I'd love to just chime in on, I agree, they all very much so marry each other, and it's and complement and go to it. And I think that number 8 really stands out to me because it does go back to the overarching of what is necessary to get across the line, right? Mm-hmm. And going back, reasonably likely surrogate endpoint expectations from full validation. Well, best criteria and best definitions are really a great starting point to understand what are these regulatory terms and what are we all aligning on, right? It's a harmonized dictionary of terminology, whether it's reasonably likely, there's examples in there, whether it's a fully validated surrogate, whether it's fit for purpose. Very important to always go back to, the basic. And importantly is, regardless, like I said earlier, if it's a reasonably likely or fully validated, you need to link the biological plausibility. And so this is where, well, how do we, as for developing a new therapy, link if, Isam, when we talk, it's if there's an inherent flawed or like with biological plausibility, and how do we make sure that we meet expectations of agency and while also meeting the needs of, patients that ultimately we're going to serve. So I think I'd love to kind of dive in more on how we can better help, developers and understanding that biological plausibility component.
Mm-hmm.
Not to put you on the spot.
Pajita, I think.
Yeah, no, I just wanted to go back to, not to just to put a quick pin on that, but I did want to go back to the point of, when we think about this framework and conceptually when we were thinking about it, right, we were thinking about the 2 factors that we considered as leveraging prior data and the uncertainty around it. But as Isam mentioned, it's impossible for us to go out and create a framework that's going to be, this is for rare diseases, because every disease will want, may need to think about it for every disease, but it's really thinking about it more holistically at a higher level. And then what are those other considerations? When we thought about our sort of applying risk weighting in that matrix approach, we started off with leveraging prior data and the uncertainty that's there, but those are loaded words. There's so much that's under there. So it's really, we need to think about how do we build that out? And that's where these elements really, I think, start to fit in, right? Some of the rare disease considerations, the digital considerations that are out there, right? When it comes to, I know with AI and digital health technologies, there's a lot moving in that space. How do those factors and considerations really fit into this? So it's starting from a point which is more holistic and a higher level, and thinking about how do we build in all of the various considerations that gets into that determination of what do we need, where do we need to focus, and then also then figure out what is that, how much evidence is necessary to get you, whether it's for a surrogate endpoint versus, a different type of endpoint that we're looking at. So I think there are many factors that it can fit into. And what we have here, sort of, that's where my thinking is we think about it as a framework, but then have like subcategories, perhaps, subs to that, to say, what are the pieces that we need to get into a larger regulatory approach or framework.
So if I were to go a little bit further into the leveraging the old data, right? So I think what we need to be mindful of is that not all data is the same. The data that we haven't generated 10 years ago and what we're generating today might be very different from an analytical perspective. All assays are not alike. So I think we need to gather the metadata, data about the data, right? So how was that assay developed? What is it that it's measuring? Ensuring that the data that we're looking at from the past is actually translatable to today's data that we're generating. Things improve over time. I mean, we've seen it in any roadmap or any biomarker development in every organization, things that we've generated, 12 years ago. And an assay might not be fitting that context of use that we're looking at today and might not be supportive of the evidence that we have today. So what we need to do is make sure we look at that. If it's satisfactory, that's great. If not, then we should have banked samples if possible. And then from an analytical perspective, repeat or generate new data, create new evidence using today's expectations and today's standards, right? Where we are today from an analytical perspective is not where we were, 12 years or 20 years ago. So I think we need to keep that, into consideration.
I'd love to piggyback off Jod because I think that's brilliant. And very tangible example is when I was leading a reasonably likely surrogate endpoint for kidney transplantation with the Critical Path Institute. Mind you, we'll get an update by the end of this year, so very exciting. But we spend years just looking what data is out there. And, hindsight is always your most important educator. But looking back there was, out of the 31 data sets, we set up data sharing agreements and to transfer data to aggregate and support there was only 5 data sets that had all the components necessary to support its context of use, or reasonably likely. All the variables to do the composite, plus that long-term outcome data. And the rate limiting step was one of the components, the analytical validation. They were not harmonized. There was only one 510(k) cleared assay available. So that was the rate limiting step for us to be able to aggregate the data. So the point is being really thoughtful upfront of what data you're collecting to then for what you intend to use it for, that fit for purpose. And this really also goes to that overarching of that prospective planning of natural history studies. NIH funds a lot of natural history studies, and there's a lot of clinical research network, but a lot of times with that data collection, and I'd love to also hear from the audience too, you have really brilliant investigators leading this, but it would be beautiful to see more involvement cross-sector for industry, FDA, helping inform what data is collected that can then potentially be used for future as an opportunity to not say all that data is going to be, and we don't need, to drain the ocean or, you know. But to have it more complete from stakeholders that may want to use that data in the future. And so I think that's a really current missed opportunity to better integrate between diverse stakeholders, especially for NIH-funded natural history studies for future uses. And so that's, you know, part of additional framework of mechanisms. How can we improve upon, right, without reinventing the wheel?
Yeah, that said, actually, we have a list of questions, you know, prepared for you all. And I think we've almost answered nearly all of them, except maybe one that I think is a perfect segue to your statement just now, Amanda. I'm going to ask you all that question. I would love for each of you to respond to this. And as I ask this question, I want to, with about 20 minutes left for this session, invite everyone in the audience, both in person and online, to feel free to come up and ask questions. We actually have a mic runner here for those of you in person. So if you have a question, you can feel free to raise your hand and he'll come to you. For those of you who are online, please feel free to continue to use the Slido function to submit questions, which we will sort through to present to our session panelists today. Now, as our audience prepares to submit their questions, and I think we already have one, which is great, I'm going to ask you all, which of the following do you think would be most useful to support implementation? Is it a maturity matrix, an archetype-specific evidence package guide, a disease area exemplar playbook, for example, some additional structured FDA feedback in the form of a template, some combination of these things? This question is part of, again, the discussion questions that you see here that we came prepared to ask each of you. So I'll stop there and see if any of you might have some ideas around that. Again, which of the following might be most useful to support implementation? Some sort of maturity matrix, archetype-specific evidence package guide, exemplar playbook, structured FDA feedback template, etc.
I mean, I'm a huge fan of examples, successful and unsuccessful. You'll see in guidance documents examples of successful biomarkers or novel endpoints, but you don't hear as much about failures or unsuccessful. And I think there's a lot to be learned. So more openness for everyone, right? All stakeholders on what went well and what didn't. And that's an opportunity to learn would be really helpful. So whether that's in the form of in guidance documents or just, you know, on publicly available or webinars or workshops, all of that information sharing without going into proprietary can be very informative across stakeholders.
Yeah.
I can go.
Great.
I'm
Yeah.
biased, but I think starting with like something that's more tangible and something that we can easily implement quickly, right? So really think about maybe the maturity matrix. I think I don't see the question here, but yeah.
Yeah, I think it might be on another
But
slide.
I think I'm looking at my notes here. Yeah, the maturity matrix, that could be a first start. And thinking about that foundational aspect of it to make things a little bit more operational, something where we can start thinking about reference point before conversations. But as I mentioned, there's a lot that needs to be built into that. And that could be a starting point, especially with, you know, Jad earlier mentioned like leveraging prior work. We have to think about whether it is even translatable from what was done before to now. And then the evolving landscape, we have how can we leverage RWE, maybe NAMs. It's building up. So thinking about that, I would say that could be a start. And then it's not just a framework, but it's like pieces to a toolkit, perhaps, right? And then thinking through some of the other aspects of what does the evidence package look like? You know, what would a playbook look like? It could be more of a staged approach, but whatever we have, I think it has to be a living document. That's really important because things are changing so quickly and so fast. So we need to be evolving with that. And then something that's easily can be used as a communication tool for us and a decision-making tool overall. I think that's important.
Yeah, and Vijita, your presentation today actually went into some detail around ways to structure the conversation around endpoint archetypes. Do you perhaps see that being part of your or fitting in some way into what it is you envision or as whatever what you just described?
Yeah, definitely. Yeah, that's kind of where I was going with the matrix that you were, that you proposed in the first one. I think it was the maturity matrix, something around that, or an element of that potentially. Yeah. But like I said, it was a small group that worked on it. So we need all the thought leaders to come together and think through that.
Yeah, absolutely. I would love to give Isam the last word maybe around this question, but Jad, I don't know.
No, go ahead. No, but there's also a question, someone in the audience.
Ah, perfect. Wonderful. Go right ahead.
Thanks. I'm happy to get the Q&A started here. I wanted to start by amplifying something, Isam, you said in your opening, and then pivot to a question. So, Isam, I think you have the quote of the day so far where you said biomarker development is hard. And I think that's very true and is probably the theme for everything that we're going to cover here today. And the other thing that you talked about is an acknowledgement that while the end goal of biomarker usage is a regulatory use, a root cause analysis would likely point to the fact that there's a lot of work to be done outside of the FDA to advance the field. And that's what I wanted to sort of point towards it and ask a question about. I think Nicole started us off this morning with a great example of a non-FDA solution, largely, to the question of biomarker development, where there was a recognition in the field of an opportunity, a broad collection of data, and then an engagement with the FDA on how to best leverage those data. So my question is, where should we as an ecosystem be focusing our efforts? The easy answer is yes, and we need the academics, we need NIH, we need the FDA, but ultimately we're all resource constrained here. So where do we think we're going to get the best bang for our buck in advancing the field? HHS has Trailblazer, which points to a little bit of work out of NCATS and some work on the Office of National Coordinator about making patient-level data more accessible. Maybe I'll just open it up to the panel. If we could sort of choose where to focus, if the focus is not FDA, right, more guidance is, where should we be looking instead?
Yeah,
Anyone?
I think that's largely a business decision in a lot of ways. I mean, companies, I'm sure, have all kinds of consortium fatigue. These are not resource neutral endeavors in terms of being part of these group efforts. But when you have that synergy between patient need, as was shown with the MRD example, is sort of clinical need. I mean, that a major catalyst was sort of the clinical community saying we need something better in terms of monitoring. So that intersection of patient need, clinical need, and the business decision, that's probably where you're going to convince, I think, companies to invest. I don't know. It seems to me everyone and their brother is involved in developing drugs for obesity and obesity-related conditions, diabetes, dyslipidemia, metabolic conditions. You can sort of start to see like therapeutic, if you want to do it by therapeutic area, you can start to see these sort of therapeutic areas where there's a lot of investment, and so it could behoove the community to identify novel surrogate endpoints so that everyone so that you have more streamlined or reasonably likely surrogates, you have more streamlined development that would benefit the entire drug development ecosystem, and then you just have to have the best drug, right? So that's one way to do it, is really see where the kind of the clinical and the therapeutic need is. The other and I think I'm making a plug for what Jad was saying earlier, is if companies are not doing this is very interesting about banking biomarkers. I mean, when I first joined FDA 18 years ago, I was in a small group that was trying to convince companies to collect this was like the KRAS days, like it was controversial, right? There were no targeted therapies. They were just coming out. Now we take them for granted. So in oncology and many other therapeutic areas, we were trying to convince companies to collect samples because these are well-designed clinical trials, and they're very good sources of sort of deconvoluting a biomarker's utility, especially if you get sample acquisition from a very large, a proportionally large subset of the patients enrolled, and you're getting pre-randomization samples. I mean, that's a huge, if that's not being done as a like an industry good practice, that might be something that, maybe there's some infrastructure that could be brought to bear that makes that a little bit more, a little less risky or less costly for companies to do.
Yeah, and along several veins that just came up, we actually have a question from a virtual or perhaps virtual audience member who submitted a question via Slido. But this question is very poignant to me because it touches on something that I bring up a lot, which is terminology, like saying what we mean and meaning what we say. And I think terminology is incredibly important within any framework or any decision matrix or communication tool, especially communication tools. So throughout the discussion, the term validation has been used repeatedly. Given that a validated surrogate endpoint represents a very high evidentiary standard, what is a better term to avoid conflating a validated surrogate with generating enough evidence to support the use of a surrogate endpoint? That's a question from one of our audience members. So I will open that up to any one of you who would like to respond.
That particular question seems like a sort of a difference without a distinction. Whether you call it a surrogate endpoint or whether you call it a surrogate biomarker or a biomarker that's fit for surrogacy seems immaterial. I mean, you still have to have that conversation about, can this biomarker be relied upon to be the primary basis for which we are making the efficacy determination on? Doesn't really, I think when you were talking about surrogates, there's no gray area. That's a fully validated surrogate. When we're talking about reasonably likely, this kind of like term of art, reasonably likely surrogate endpoint, that is to say, a biomarker that is, and I'm just focusing on biomarkers, a biomarker that, is reasonably likely to predict clinical benefit and is the basis for an accelerated approval, for example. That's what we use those for. That's where terminology gets a little tricky. Reasonably to whom? And maybe this is where a framework comes in. The dimensions of reasonableness, right? There's no statistical definition of reasonably likely, and there's no biological sort of mechanistic definition of reasonably likely. And so that could be whether we call it a maturity matrix or an evidentiary kind of communication tool. I think that's where we can start to like bring in a little bit more granularity. But I totally agree with the person who asked the question around, things like exploratory biomarkers. I mean, I don't know what that actually even means. And I can tell you as a reviewer, if I see a sponsor submitting an exploratory biomarker, like I have to prioritize other evaluations. And so if it's not clear what the biomarker is intended to inform, either in terms of downstream clinical development or regulatory purposes, approvability, labeling, so forth, then I don't know how to relate to that proposal. So that might be something that also needs to be clarified within an evidence package. How are you explicitly trying to use this biomarker for a decision? What is the decision you're trying to inform? How do you want regulators to look at this?
And that resonates recently in multiple panels or discussions I've been on, talking about rational-based approach, right? I think that's where you're going to, like, what does exploratory mean? So going into details, I think from the sponsor side, what we can do a better job with is explaining that, right? What's the thinking? Why did we choose this approach versus another? Like showing our work. If we're doing a math problem, we're asked to show our work, right? Not just the answers, and not just saying, oh yeah, this is exploratory, to this example, right? But really being able to show how we got there, what other considerations were made, why didn't we go a certain route, and why did we choose this? Because sometimes things get lost in that if you don't do that. Because there may be an instance where FDA may come back to us say is like, oh, you actually decided not to go that route, but we may want to re-explore that, right? Maybe that's something that needs to be considered. But yeah, those elements, I think, thinking through, you know, as we think about this framework and the package, maybe there's like some considerations around how do you show your thinking, as well as if we think about a toolkit more broadly.
I was going to add that regulatory science is its own language. And many ways to go through it is through examples. It is not intuitive. It is not easily understood, even for those that have been in the field for a long time. And how we define something evolves over time, and how it's interpreted is the translational part. And so there's an art and a science to applying basic science to regulatory strategy to that translational aspect. And there's and this is really where, regardless of your experience or knowledge or scientific background or regulatory background, going through examples and walking through those of where there's been alignment or where this is how we did it, like Nicole's with the MRD. This is how, this is where there's that unmet need. This is how we align. We need a standardized MRD, the assay, and pulling in is a really tangible way to translate, like, what is this definition and how do I actually apply it to what I'm trying to do? So one of the parts is even within the agency, that education and that teaching, and everyone in this room, if I ask, what's the definition of a biomarker, we're going to get different answers, right? And so there is an importance of information sharing and knowledge sharing for all stakeholders, including within the agency, because then it helps them also align internally of how they go through and interpret different packages and the evidentiary associated to it. Reasonably likely surrogate endpoints. It's not a terminology with the European Medicines Agency, right? And so understanding where you can have more alignment when you're trying to think of a novel endpoint and get it regulatory endorsed. For international clinical trials, it's now navigating, those different complexities when you don't have the same language. So I love that question because it is always going back to basics. Like, what is the minimum necessary to get across line, and how can we really make sure we understand the precedent, and regardless of therapeutic area, to help inform what's that totality of evidence?
Just quickly want to add, you talked about examples, and I think examples are really important, whether it's coming from both from the FDA side, right, the regulatory examples that we see, and I think the leader, forgetting on the full day, the leader examples are great. We're learning so much from those, more of those, but not just, the onus isn't just on the regulators, right? We as sponsors need to do a better job of publishing as well, and putting out our examples of what's worked, what hasn't, and if we can be more open, or some of the work that has been done, right? Just putting it all out there to show what was the thought process, how did it start from X and lead to success at the end, right? Getting those examples out there, publishing on that, I think would help as well, so that we as a community can learn more and learn from that. overall.
Yes, wonderful. Well, we have a minute left, and I can probably summarize a few things. Oh, perfect. Okay. Okay. Well, perfect. All right.
I just wanted to touch on a comment that Amanda made. When you said the phrase regulatory endorsed, it kind of set off some bells in my mind. So when you're pursuing a biomarker for an individual drug development program, and you might think you have alignment with the FDA, in your early stage clinical trials, or even beginning a long running phase 3 trial, you can have subsequent conversations where the goalposts shift, right? And that creates a huge risk for sponsors. And so maybe building in that could be part of the framework. Like, how do you know when you have that endorsement and are, ready to go full steam ahead, and pursuing that full evidence package or committing to that as your primary endpoint?
I don't know if there's going to be session on this later, but it's the earlier the better in terms of having those conversations. And I would say two brief things. One is I wouldn't just look at it in terms of biomarker. I would look at it in terms of biomarker and context. I'm thinking about like bone mineral density, which has been successful in some cases and failed miserably in other cases as a function of the disease, like what we're talking about in terms of the disease pathogenesis and the pharmacology and how that modulates that disease. So I think we should always be thinking about the biomarker in relationship to the pharmacology and the disease biology. So that's just one kind of housekeeping point. The other is on this early conversation about getting regulatory endorsement, lessons learned, and transparency. One of the things that my colleague Mike Pakinowski talked about this earlier in the year, one of the things that would be that is very helpful for us when we're doing evaluations is we need to know what the counterfactuals are and what the uncertainties are. We're going to look for those anyway. So if we have an evidence package that's biased, that is excluded, like all of this data that is not in support of the biomarker, it creates a situation in which like maybe a reviewer doesn't want to do any more work. I mean, like you haven't presented a full picture. On the other hand, if you present the full picture and you say, and here are the counterfactuals and uncertainties, that's a strong starting point for a conversation. Then we can talk about how do we think that those inconsistencies are relevant? Is there some sort of good reason why? But is that going to completely discount the biomarker? I understand that companies think that's probably a risky proposition to sort of like present all the data. What a radical idea. But we're going to do that anyway. Our thought exercise is to completely vet the claims around the biomarker, including comprehensive evaluation of what's not presented in the regulatory submission, what's out there in the literature. So that I would say that is a good, I know it's risky, but that's a good practice that I think we need to get over and start having those conversations early as well.
Wonderful. Well, we are slightly over time, and we are standing between you and our lunch hour. So I'm going to conclude our session and thank each of our panelists once again. Fujita as our presenter and panelists, thank you all for being here. And this has been a fantastic discussion, and we look forward to doing more of this after lunch with you all.
Okay, hello everyone. Welcome back. We're going to get started in just a moment. I think we're a little late coming back, but I'll reintroduce myself in case anyone is just joining us for this afternoon session. Hello again. My name is Valerie Parker. I'm an assistant research director with Duke Margolis on our biomedical innovation team. So as we move into all good water. So as we move into session 2, we'd like to shift from discussing evidence maturation towards a conversation around the shared infrastructure, incentives, and evidence reuse mechanisms that can make biomarker development more feasible and scalable across the broader drug development ecosystem. So more specifically, panelists will discuss the role of pre-competitive organizations, public-private partnerships, sponsors, regulators, patient groups, clinicians, and researchers in supporting shared investment models, disease community alignment around biomarker priorities, validation infrastructure for complex biomarker tools, and data measurement, harmonization, and governance approaches for data reuse. Panelists will also discuss additional expert and audience input on how those ecosystem-oriented recommendations can be refined, prioritized, and implemented. And we'll be bringing Slido back again. So to kick this session off, and move to our next slide. Oh, it's already there. Great. So to kick this session off, I'll give a brief intro to our panelists, and then I'll ask them to share their initial thoughts to frame our discussion. So first, we're joined by Ramona Belfiore Oshan, the Executive Director of the Duchenne Regulatory Science Consortium at the Critical Path Institute. Next, we have Darcy Fear, Director of Global Regulatory Policy at Bridge Bio. Next, we have Nicole Gormley, the founder of Gormley Advisory, and who we heard from earlier today for that use case presentation. And finally, we have Steve Hoffman, the Senior Vice President and Chief Preclinical Officer at the Foundation for the National Institutes of Health, or FNIH. Ramona, I'll turn to you first for your opening remarks.
Sure. Thank you so much to the organizer for the opportunity to be here, really representing the Critical Path Institute and in my role in advancing the biomarker development for rare diseases, but also bridging to other rare disease and non-rare disease areas in the neurological space and not. So from a perspective of biomarker development, really my remarks wanted to point out the differentiation between what we mean by biomarker discovery and how we go into actual development of a fit-for-purpose biomarker. Many organizations invest and contribute to biomarker science and advancing the evidence that support the biological understanding of certain mechanisms. But how do we get to actual utilization of these biomarkers in clinical trials require a whole set of infrastructure and standardization or harmonization of the data that we continuously work and try to emphasize in our pre-competitive consortia as neutral convener of these solutions. So our role is really to bridge the gap to between the question of is this marker going to be significantly and scientifically adequate to how do we use this biomarker in advancing drug development. Consortia can certainly play a big role. So in advancing these developments, I think the collaboration with all stakeholders, really timely getting together in one single space to share the uncertainties is even more important than what certainties we have. And so what we're trying to do is really reducing that scientific uncertainty that bridges the knowledge from preclinical research to clinical research and to adoption, which is the end goal that we are trying to achieve.
Thanks so much, Ramona. And now Darcy.
Hi, so I'm Darcy Freer, although I like the name spelling on the slide for Fear. I just noticed that. I'm at Bridge Bio and we focus on rare genetic diseases. And so biomarkers is a big passion of ours to try and figure out how we can best advance research in this area, advance rare disease drug development. One thing I wanted to touch on is a lot about the pre-competitive space and making sure we can align better as a group of stakeholders to find acceptable biomarker evidence. And I wanted to share a brief example. We were part of a workshop in 2024 for limb-girdle muscular dystrophy, scientific workshop that built upon a PFDD, a patient-focused drug development meeting, that really mapped the patient impact. And they just thought a natural next step would be to go dive more into the science and bring together academic experts, patients, caregivers, advocates, senior FDA leadership, and of course, commercial developers to really talk through this. And some themes that arise were like limb-girdle muscular dystrophy pathophysiology is pretty well understood. And tailored causal pathway approaches are now feasible for various subtypes of this dystrophy. It really then ended on a totality of evidence approach, like muscle biopsy biomarkers could be possible candidate surrogate endpoints, or single arm trials plus natural history data, or platform approaches are all things that we should consider and definitely are at the right time to be able to consider those. And what it yielded at the end was this next step of being able to provide scientific leadership the opportunity to kind of illuminate this pathway forward. And it really emphasized the importance of aligning with patient advocates really early on, because they're very important. Their voice is very much highlighted, especially by FDA. So it's something that we were really enthusiastic about. And this is an example of what a pre-competitive space could look like for future biomarker discussions and ensure alignment for a path forward. And I'll pause there.
Thanks for highlighting that, Darcy. So we'll go to Nicole next.
Hi. So I've already shared a little bit about my perspective and experience, especially as it relates to minimal residual disease and multiple myeloma. But I would just add a couple of, or highlight or underscore a couple of aspects that I think are really pivotal in any field when thinking about biomarker evidence and further development. And just to highlight, I think there's a couple areas where I think that the recommendations really can help advance how we approach this. Is just, I think it's really important to have some standardization. And I think looking back on some of the examples I've seen over my career, the standardization of the field, socialization as well of a biomarker within the larger community, such that there is some standardization, some harmonization, such that the information is collected in some uniform manner to allow for greater applicability and use in other settings is pivotal. And that really sort of requires a lot of community alignment and community engagement. And I think, we've heard throughout the morning some of the comments just that this is a really heavy lift and it can be hard to do in isolation. And so there is this tension of engagement in a pre-competitive space, but then also sort of recognizing the unique incentives that are available to one specific organization. There is this tension, but so much can be gained by collaboration and having consensus, at least on some of the areas as it relates to assay and testing parameters that there's so many advantages, but it does require a lot of community involvement and ecosystem involvement to sort of get that alignment.
Thanks, Nicole. And Steve, we'll turn to you.
Thanks, Valerie, and also to Duke Margolis and the folks here for having FNIH here at the table. Obviously, we are known as the official partner of the NIH in support of their mission, but a lot of that effort is through public-private partnership as a neutral convener to really echo what was been said this morning and said here. To bring cross-sector partnership together to really solve some of these challenges and problems, whether it's within a disease, across disease platforms. But by far and away, we've been keenly involved in a lot of the biomarker science that's been done. When I had kind of interrogated that policy roadmap, it stands out that we kind of moved beyond discovery of biomarkers as a problem. It's really about the evidence infrastructure, the investment problem that we have. We have extraordinary scientific opportunity, but our ability to validate, standardize, share, and reuse biomarker data really has not kept pace. And we really don't have the funds and support within individual efforts to do that. It's pretty remarkable when you think about that because modern medicine depends on biomarkers, and that's been restated by many from here. They guide diagnosis, prognosis, treatment selection, and obviously increasingly regulatory decision making. But while the therapeutics get all the attention, they get all the press, they get all the dollars, it's the biomarkers that enable those decisions, but that remains largely unfunded. We have lots of tools, and a lot of times we talk about where we're frustrated with FDA or where we don't have resources or guidance. I would remind us that there is FDA's Critical Path Initiative, FNIH's biomarker qualification efforts, and the biomarker qualification effort of the FDA, the Critical Path Institute, and what CPAT does, and our own biomarker consortium and the BEST framework. There is guidance out there. There are guiding publications and information, but it's fragmented. It's out there in the abyss, and we know with our phones and with everything we do, what was published yesterday is gone from our mind, gone from our guidance for what we do. So hopefully that maybe some of our efforts in the playbooks that you hear us talking about, we can kind of revet in 2026 and beyond that type of information. Really understand where the fragmentation, inconsistent standards are out there, governance barriers, limited incentives for generating this type of evidence. And I particularly want to also, and I'll speak to this perhaps in some of the questions, talk about historical. Jad made some comments about historical biomarker sets. Not only is technology and AI machine learning now upon us, that we can go back and interrogate these data in a different way. Can we go and get samples and do new testing and bring them into this differential use under their original context of use? I'll be the first to mea culpa. In our biomarker consortium projects, there's about, over its 20 years of existence, about 45 different projects. There's about 45 different data sets somewhere in the world, academically or in a commons somewhere, where we might have access to that data, the metadata, samples that could be used, but we have not done a great job of bringing that back into the next steps or the further evidence generation. So I think my call to action is simple, that we need to think beyond these individual projects, invest in durable biomarker data commons, sample commons, validation infrastructure, and public-private partnerships to get this done. So that evidence generation has to be viewed as foundational scientific infrastructure, not a byproduct of drug development. And if we can build those right incentives, the right incentive models, governance, and biomarker science could certainly accelerate precision medicine for decades to come.
Thanks so much, Steve, and thank you to all the panelists for, I'd say, previewing what our discussion is going to be about and giving the audience a sense of your different perspectives that you provide. But audience, we want to make sure that we're getting you involved too. So we're going to bring up our new Slido poll. I can get the clicker going, where we have recommendations under Goal 4 for our policy roadmap. So just as in the prior session, you can join Slido through the QR code or by going to slido.com and entering in the hashtag bio, BIO, into the line there. And among the list of the recommendations provided, we're asking that you select 3 that you feel would be worthy of further discussion or consideration. And so I'll go ahead and read through these different recommendations that we have outlined in our roadmap as you all are making your selections. So for the first one, or number 35, public-private partners should explore incentive or funding models that reduce the first mover burden of early biomarker evidence generation, especially where evidence assets, assay standards, or analytical methods could benefit multiple product programs. Next, FDA, NIH, FNIH, patient groups, clinicians, and industry should work through neutral conveners to align disease communities around shared biomarker evidence priorities. The next recommendation, FDA and public-private partners should seek stakeholder input on how evidence and regulatory learnings from individual submissions can, where appropriate, inform biomarker qualification, context of use expansion, and broader shared evidence generation. Our next recommendation, public-private partners should support validation infrastructure for complex biomarker tools, including AI-enabled pathology or imaging, biomarkers derived from new approach methodologies, and multi-component measurement approaches. The next recommendation we have here, public-private partners should support standardized cross-trial data collection for high priority biomarkers that require evidence across sponsors, products, and populations. For our next recommendation, public-private partners should support shared natural history data sets and longitudinal biomarker data sets that can help link biomarker trajectories to meaningful clinical outcomes. The following one is prioritize data harmonization, assay standardization, and common measurement practices before pursuing high-stakes surrogate endpoint acceptance. And I believe this is our final one here, design protocols, consent language, and governance plans, including data use agreements, to retain and reuse exploratory biomarker samples and data after trials conclude. So it looks like we have about 35 people on the poll right now, and the number is growing a little. So I'm going to give you all just a moment to make your selections, and then we'll see what that ranking is looking like. Now that we have around 50 or so participants, let's see how we're looking here. Okay, so we have a tie. That's interesting. So right now, the top recommendations that are coming in. Oh, I suppose you all thought we couldn't have a tie here, so make it easier on our part here on stage. So right now, what I'm seeing is recommendation number 40. Public-private partners should support shared natural history data sets and longitudinal biomarker data sets that can help link biomarker trajectories to meaningful clinical outcomes. And then also, still really neck and neck, although again, as I highlight it, you all bring me right back down to earth. Recommendation number 36, FDA, NIH, patient groups, clinicians, and industry should work through neutral conveners to align disease communities around shared biomarker evidence priorities. So again, bringing that back up neck and neck. So I wanted to turn to our panelists here and see what are you thinking of, especially these first two that are, it seems like everyone agrees that they should both be first, potentially. Perhaps, Ramona, I can start with you.
Sure. This is really in the rally of what the role for Critical Path Institute is in driving this communications and fast collaborations for advancing the evidence, but then developing packages for regulatory submissions and the adoption of these tools in clinical trials. And when we talk about the sharing investments and the understanding why these public partners can be powerful, one evident example comes into mind, really goes into consortia. There are long time standing collaborations where really, because of the stakeholders' knowledge of the needs and the unmet needs, we are able to provide stronger assessment also in communication with regulators on the unmet needs, and so work together towards a specific goal. And one very tangible example has been the collaboration with the Critical Path Institute for Parkinson's, where there was a long-time collaboration with the Michael J. Fox Foundation, the FDA, and the example of the alpha-synuclein seeding assay letter of support as a push for advancing drug development in this space. Really, there was an early investment in the assay development that was supported by the advocacy group, in this case playing a very strong role and really leading the path for the alignment. And this really is a testimony to the importance of having patients telling us what's relevant in the space and driving that innovation piece for alignment across the community. Through that investment, really, there was the realization of data collection that was harmonized, and only throughout the composition of large-scale natural history data that were compiled through the consortium activity and the early alignment with the FDA Office of Neuroscience, the letter of support took only 3 months from the submission to the posting, based on the level of evidence and harmonization and standardization that was supported. Another example is bridging the gap from preclinical evidence to clinical evidence that we've seen applied with safety kidney biomarkers that were FDA approved in a panel that really is being utilized in more than 150 clinical trials. But only after the endorsement from the FDA, we've seen in 2 years 40 approvals and adoptions of these actual biomarkers in clinical trials. So what we've seen is the incentives, not just in creating evidence, but the more adoption is applied, the more sponsors feeling comfortable in including these measures in clinical trials. And this is what I think is really relevant in pushing forward the actual advancement of the drug development process.
Thanks so much for that, Ramona. And I do want the other panelists to weigh in, but since it's so applicable to your points, I wanted to make sure I highlighted the third one on the screen as well, which is our recommendation 41. Prioritize data harmonization, assay standardization, and common measurement practices before pursuing high-stakes surrogate endpoint acceptance. But I really would like to turn to the other panelists and hear your thoughts on these top 3 options that we had.
I can jump quickly in for point 36, just because that was one of my top choices about having neutral conveners. And I think this even comes to one of the questions that we have here about lowering risk of collaboration with others to reduce that first mover burden, just because if there's a trusted neutral convener, that can really help define what biomarkers are the shared priority. And then it also kind of gives an incentive. We have different groups that are going to this. Your competitor is deciding to attend this meeting. This patient group you're working with is attending this meeting. You're more likely to come and join that conversation pretty early on. And it just becomes a more attractive path forward. And I also just wanted to say that definitely there needs to be clear actions when it does come to some of these neutral convening meetings. At the end of a workshop, there needs to be some sort of clear next steps in order for people to see the value, for the stakeholders to see the value, and an incentive to work together.
Thanks so much, Darcy. Any other thoughts from Steve, Nicole on these top 3 recommendations?
Yeah, I think I emphasized already the 41, just the importance of harmonization, standardization, and then also 36 as well, the importance of having alignment in the broader community, that it is really resource intensive and there are competing interests. And so if there is within the broader community, by whichever means, some consensus that this is where additional efforts are needed or efforts should be focused or resources, that can help the process. It does require a large ecosystem of involvement, though, and it's hard to place the responsibility for development of biomarkers squarely in one camp. And although there's a tendency to do that, it really does require, sort of a village coming together to have alignment on the priorities where the need is, and then also just to have adoption of any standardization or harmonization that is put forward. Sometimes that's a very hard ask.
I was pleased to see that this is what the attendees in the group really came to. We didn't cheat. Ramon and I did not have our organization send in responses to the poll, but it spoke to public-private partnerships and what CPATH and what FNIH are really about, and getting that information out as quickly and as openly to the public to move the field and all boats is really core to our efforts. You talked about the trusted conveners. I think both our organizations have been very successful. We have a little bit unique ways that we work, but in particular, Ramona brought up the Kidney Safety Project, work where we're doing in the NAMS field or whatever. We're actually doing that collaboratively. And so there's even, I would even say, a double or a multiplier there where we're able to bring in multiple organizations that has a remit, their strong ties to FDA, their access to some other international data, and the kidney effort in particular enabled us to further validate what we had came up with in a panel. The other thing I would tell you that if you get that joint expertise in the multiple cross-sector ecosystem we're talking about, you often come out with not something that benefits the one, but obviously benefits the all. When we think about some of the successful biomarker qualifications, they haven't always been in a particular area. The kidney safety markers in particular, really apply to all safety, clinical safety markers. When you get into the surrogate endpoint efforts that we've done in osteoporosis, yes, absolutely is around fracture and osteoporosis, but really having a roadmap of how to get to surrogate endpoint. But then the third one on 41, the assay standardization harmonization, Nicole and I were even reminiscing about it at lunch. That effort started really as a prognostic biomarker and really getting all the disparate types of data, harmonizing that, standardizing into it. But it was the joint expertise, the alliance, and working with the FDA that really became that an accepted endpoint and that acceleration endpoint. And she even noted in her slides that is likely really been well adopted and utilized and been successful. It doesn't need to go to qualification. It's really became the standard in the field. So I think that type of public-private partnership, that type of harmonization, when you get joint expertise together and really understand what is the immediate unmet need, what is the pathway, what is the right context of use in question, it powers those types of partnerships truly power a good decision in alignment with FDA to get to our goal that we're trying here today.
Thank you both for those insightful comments. I was curious if we could scroll down to see if there were any other recommendations that were being highlighted or what that ranking looks like. Okay, so we have another tie between our recommendation 37 as well as 42 relating to FDA and public-private partners seeking stakeholder input on how evidence and regulatory learnings from individual submissions can inform biomarker qualification, context of use, and then also around designing protocols, consent language, and governance plans to retain and reuse biomarker samples there. And then if we could keep scrolling just to so we're all on the same page here. I think you can also scroll on your devices as well to get a highlight of that. So I'd say it's pretty even across the board, but especially at least we had those top two. I wanted to turn now. Oh, there's even more. So I guess I'll highlight, and then the one that's coming in sort of at the bottom here is number 35, related to incentive or funding models, which just closer around 30%, still close, I'd say, to reduce the first mover burden of early biomarker evidence generation. But I wanted to turn now to our discussion questions that we had prepared, and then also remind you all that you can submit questions via Slido, and then later we'll also have mic runners around as well if you have questions. But wanted to highlight a couple of questions that we had for this session. So perhaps this is a great tee-off actually with highlighting the funding models, which is what shared investment models are realistic for reducing first mover burden, particularly when biomarker evidence, assay standards, or analytical methods could benefit from multiple sponsors, but the near-term incentives could be uneven. So would any of our panelists like to start with that question?
So, Melly, I did want to state, I was a little surprised. I get that it's, 30%, 28%, but the investment, I think, is an important piece. And having novel investment, it's not going to be biopharma that's going to do this. There's going to be some self-interest there. They're going to be part of the larger picture, but I don't think they're going to take it on individually. Obviously, smaller biotechs and other developers or providers don't have really the dollar or the bandwidth to take some of these efforts on. So that's why the partnership model works real well. We have begun to explore and push buttons as best we can through the federal government, maybe the ARPA-H program. I certainly could echo many of Janet Woodcock's speeches and soapboxes where she talks about that investment from philanthropy, investment from even venture. Where are there unique opportunities where we can sell together that message? We were talking also earlier, let's say in the NAMS model, where we're replacing some type of existing biomarkers, assays, or studies. That can be done in the pre-competitive space on the front end, but ultimately put people at the front of the line for investment in those technologies. And that's where venture money may be. But can we make an offer? Can we make that joint expertise to say we need investment early on for the whole to get it to the point that you would then have some prioritized venture interest there? Same with philanthropic efforts. We need to start going to these family foundations, these larger philanthropic organizations, and explaining the biomarker as we had all talked about earlier, how essential that is. It's not about drug A, drug B, but it's about the infrastructure, the assay harmonization, the standardized efforts, and that their money is highly valuable and very much can make an impact in the field in that way, which is often what they're looking for. So find those types of other shared investment, I think, is an important thing that we have to change what the old thinking is to what the new thinking needs to be.
Yeah, appreciate that, Steve. Just really highlighting that evidence generation is expensive. So figuring out these investment models, funding models are certainly important. Ramona? Steve?
Sure. Well, I guess, Steve, what we have seen is that the shared investment model, what works best usually is when participants can see a pathway from the evidence generation to the practical implementation of what we are creating. And so explaining the impact to the large scale of how these biomarkers can be applied at all levels. Sometimes it's not just looking at the regulatory pathway, the application of a drug, but it's also investing on having a tool that optimizes the clinical trial design, which is indeed de-risking a lot the output that you have in the long term term for your successful in the program. And so investing in measures that and quantification of these measures, maybe utilizing a biomarker in disease progression models. We've done this in the case of Duchenne muscular dystrophy by utilizing imaging-derived biomarkers and characterizing the longitudinal trajectories of those to describe how the disease changes. And that helps inform the trial design, and sponsors now can feel more comfortable in defining potential thresholds that help assessing the ideal population or the adequate population for the specific target approach. So what we think is certainly, again, having multiple goals and having a specific scenario that we can present to those that invest in the development so that we can advance the discussion for investment and shared learnings. We've also seen a case for increased investment when we have the ability to move across multiple contexts of use. For example, we have participated in a collaboration with the Predictive Safety Testing Consortium at CPAT into contributing data for the approval and the qualification of GLDH as a drug-induced liver safety biomarker. And this is going to impact not just the community where we see the implementation of this biomarker in more broadly, but in the specific, the use for the muscular dystrophy community when you have underlying muscle damage that will be indeed confounding the results due to the elevation of transaminase in the liver. So it's the adoption of these tools across multiple communities, addressing issues that might be relevant for rare disease communities that do not have a natural history data that can support the development of biomarkers that I see strong potential for motivating the initial investment and all those burdens for the long term.
Thanks, Ramona. Darcy or Nicole, any additional thoughts here?
Nothing too much to add other than I really liked your point about de-risking and making things more predictable for sponsors. And I feel like that is something that would be a way to just incentivize doing things like this in general. Yeah.
Great. Well, then perhaps we can move on straight to our next question that we have here, which are, what are the real legal, contracting, consent, privacy, intellectual property, and competitive barriers to data and sample reuse? And which of these could be addressed through template agreements or earlier protocol and consent planning? Would any of our panelists like to begin there?
I mean, I'm happy to jump in, but don't want to steal the voice. I think there's a misconception that like data reuse is often a legal problem, right? Much of it is really a planning problem. The old adage of failure of the plan is a plan for failure. If you're not thinking about future use and your consents, reusable governance, standardized agreements, and kind of these interoperable data standards, then you're having an issue. I'll give an example. In our accelerating medicines partnership, we have this across various diseases from autoimmunity to AD to PD and others. These data sets and these multi-omic data sets and all the additional testing and imaging data, it was not all standardized from the beginning. And now we are faced with trying to look across these different portals of data. And we have a program that we're looking to launch in the near future on a systems biology of inflammation to look at these inflammatory profiles and multi-component markers that may suggest progression. And it needs to think about, the metabolic side of it and the autoimmune side of it and the neurodegenerative side of it. And this is really what biology looks like. And we have to do that. But if I already did not pre-plan that these data sets and data commons and how the data and how it readouts are going to be similar, harmonized, standardized, as we've been reiterating, it's going to be challenging, even with the power of machine learning and AI. So we have to really be thinking not just what's the future of 2026 in the second half of the year, but what emerging technologies, what emerging type of data types do we think are going to be part of these or could be enabled so we can do such a cross-disease type of analysis.
Ramona, please.
Yes. Well, in looking at the burden, I think what Steve highlighted is very relevant. The fact that beyond the legal issue and the ability to share data, then there are other elements to consider really in the quality of the data collection. And sometimes when we try to compare biomarker data, the pre-processing element is so fundamental, and going to compare different studies, realizing that the biomarker data collection at the time during the day in the population where those elements are so relevant now become the reason for not being able to compare study through study. And especially looking at targets like, again, skeletal muscle injury. In one study, there might be data collection that is looking at making sure that there is no activity the day before the data collection, and some others that does not become relevant. So how do we work together to make sure that there are protocols for pre-analytical assay harmonization and all the post-processing harmonization, all the elements that go into making sure that that information becomes then reusable, really goes beyond the legal challenge, which is certainly a very relevant one, but in the context of biomarker development is one of many other pieces, and the analytical validation step being one of the most challenging, I think.
Thanks, Ramona. Any additional thoughts from the panel on this question? Well, in that case, I wanted to shift to the room. We do have a couple mic runners around, see if there's any questions in the audience. And then we're also soliciting questions online. But I also have a few more questions for the panel here. So as we're getting our mics up in the room, a couple more for consideration, and then I believe we'll just kind of keep going in order here. What should sponsors, consortia, and patient groups build into protocols, consent language, and governance plans at the outset, so exploratory biomarker samples and data remain reusable after trials conclude? So wanted to get the panel's thoughts there.
I think, and I won't comment on, the legal or other aspects, but just maybe underscore some of the comments that were just made in terms of ensuring from a clinical and analytical standpoint, that the information obtained in any trial or study is actually subsequently, usable for other, analyses or pooling of data. And perhaps that harkens back a little bit as well to the idea of having some broader consensus among the community in terms of how and standardization, harmonization of not just, the assay, but, some minimum thresholds or minimum criteria for harmonization of assays, but then also other criteria related to collection and sample timing, and some of the other examples that were mentioned can have really large impacts. And it's one of those things that there are opportunities for, that obviously we can't even begin to foresee now, but there are opportunities to look back at data and to have some flexibility in terms of, well, it may be appropriate to pool some populations, or may be appropriate to look at some therapeutics when looking at a meta-analysis, or some group or large data or data sets. But if there's too much heterogeneity in the information that's included, it can really limit our ability down the line to draw any meaningful conclusions. And so it's really hard at the outset to sort of envision where we want to end up. But having some sort of alignment, if possible at all, regarding, the collection procedures, timing of assessments, and some standardization regarding assays, if possible, can really go a long way.
Appreciate that. Steve, you look like you want to weigh in a bit.
I was just going to kind of echo something that really kind of rang a bell of an existing program where we're looking in neuropsychiatry into depression and looking at multimodal markers of depression to understand and subtype and phenotype patients. The clinical, not only does that need to be a common methodology, a common standard way that it's done. Everyone and multiple companies have successful, even with the FDA, have done their own thing, and they use this cognitive measure, or they use this imaging measure, or EEG, or whatever it may be with a particular company. But the idea of having those clinical workflows in particular aligned with what is appropriate. You can't be doing your imaging on day one and 3 days later you're doing a PRO, asking them questions that's directly related, and particularly in particular for a particular disease that or their condition that changes. And so there needs to be a harmonization and standardization of those types of things, including the clinical workflow. We need to start applying those. And then the second thing that I would think about, and maybe ADNI is still the great model of you have to upload that data that you produce by using ADNI samples back into ADNI before you're able to interrogate or go further with that. Even at the critical path, the BMDR is really designed for CDIS level type of effort. Maybe we should be enforcing and pushing that. Do we need incentives? Do we need dollars to upload that data? But maybe the partnerships that we do, CPATH ourselves, they do it much more than others. Maybe that's the repository that needs to be there to help accelerate an expanded context of use or a reuse of that type of data because of that repository is actually there. And we have some of the tools, some of the capabilities upon us, but we don't utilize them or we don't push them or we don't require them. And maybe we should be requiring those types of utilizations.
Thanks so much, Steve. Darcy, Ramona, want to add on to this discussion?
I think only I from what both of you are saying, I think building in sort of, and I think you've referenced this, but building in a role for those neutral conveners from the very start, to think about like what kind of questions and discussions need to happen in order to make sure everyone is aligned as things move along. So I thought I agree with what you're saying. That was something I would add too.
Great.
Well, addressing further, I guess, the reuse of this data, I think it will be really helpful to focus in future use consent language, and also to just understanding how we can implement better sample retention plans from a sponsor perspective. When a clinical trial fails, then we see sponsors closing up the team and kind of dropping the follow-up steps that might indeed allow for reutilization of the samples, or even pre-specifying that that sample will be useful for future research, not associated to the immediate program goals, should be pre-specified by the sponsor. Otherwise, those samples unfortunately need to be discarded and lost. And that's, as we know, loss of resources, investment, and good specimens from patients have been invested a lot of time and their families. So again, going back to the concept of patient-focused drug development, how we can make the best of the investment that patients have made by participating to the trials, by really being part of the research, and how we can communicate better back to the patients that biomarkers are indeed going to be relevant for not that specific research only, but any future research. And so that's why they should consent for further data sharing and specimens sharing. So that's my hope for the future there.
Thanks, Ramona, and appreciate you highlighting that patient perspective piece as well. Wanted to turn to the room to see if we had any questions in person. If not, my team is sending me a couple of questions from Slido as well. So I'll begin with the Slido questions then. So building on the panel's discussion for the need for shared investment models beyond individual sponsors, which stakeholders, in your opinion, are best positioned to make the case for and mobilize government, philanthropic, venture investment in biomarker infrastructure? Look at you, Ramona.
I can start. Well, my position, I think there is no one single sponsor, one single entity in the community. It really needs to be coming from all, as a community effort from all perspectives. And understanding how we can do this in a true multi-stakeholder effort. It's not up to the FDA to prescribe what the assay needs to be utilized. It's not up to the sponsors to only make decisions around what's adequate for the clinical trial, but it's going to be up to the entire community, and reemphasizing the role of patients, including patients in understanding what's the burden for collecting biomarker data, and that feedback being included into the development of biomarkers is certainly relevant. So in terms of understanding how we align all together, again, having a pre-competitive effort is always what brings us farther, not faster necessarily, but certainly farther.
Thanks for that reminder on the importance of collaboration. And would any of the other panelists like to add on? Thinking through which stakeholders are best positioned to make the case for or mobilize government, philanthropic, and venture investment in biomarker infrastructure.
Needs to be in combination, right? I would tell you that I don't think our organization has effectively fundraised or sought support without kind of that joint voice. Obviously, the federal government is not going out there asking private sector for dollars, but they show the importance of the basic research that needs to be there. And they're the first part of the call or the first part of the outreach that we have. We then bring individuals from the private sector to support what that application and how they will use that to drive drug development. But then there are going to be pieces like primary care or a physician or an MD that kind of explaining how this can quickly translate into clinical care, change practice, change standard of care. And then obviously, probably maybe what I should have said first is where the world has changed to is you need those patients involved in that story, that I understand this, that I understand how this benefits of me. Yes, I want better treatments. Yes, I need something now. Yes, my family member is, challenged by this, or it's a rare disease, and I really want to focus on my disease and my child or my family member, but also they can be just as strong if we can help educate them about what we've been talking about with biomarkers and the infrastructure that is really important. If we can get that same voice out about what we're talking about with those same groups, I think you might be able to see a greater investment from congressional dollars. You might be a better language that goes out. So you might see an ARPA-H type of effort on biomarker validation and biomarker qualification. You might see the philanthropic topics that we had talked about, and I might have mentioned, and others have definitely mentioned, as investing in this type of area. You have to still build the building and lay the concrete and the foundation. You can't just get the pretty building and the windows and a great stuff. You need the girders, you need the concrete to build those types of things. We all need to message that in a joint way, and we need to help educate our communicators of who's really getting to funding sources from individuals to organizations.
Thanks so much, Steve. Any other comments on this question?
I just wanted to follow up and say I totally agree that it's like a joint community effort. And it might depend on who starts those conversations too. Like for the workshop I referenced, it was a patient organization that really kind of spearheaded, but they are the ones who are able to talk to the various groups and get alignment of like who should be in the room. But definitely agree that there needs to be that collaboration for this to really be successful.
Appreciate that. Any other comments? If not, we are getting some more questions from Slido. So one question that we had was, how can stakeholders maintain consistent assessments within and across longitudinal studies while incorporating scientific and methodological advances without increasing protocol burden for patients? I think I'll read that one more time, just so we all have time to digest that one. So how can stakeholders maintain consistent assessments within and across longitudinal studies while incorporating scientific and methodological advances, all without increasing protocol burden on patients?
I think there is, I'll start. I think there is a balance there, right? The tension between innovation and then also consistency and harmonization. And I think perhaps, again, that it is sort of where the broader community or, broader voices from the ecosystem can play a role. Because you do have to prioritize. You can't have your cake and eat it too. You can't do everything, right? And so there is a role, and we need to continue to foster innovation. And sometimes that will be in a private manner, where some companies benefit more than others. But by the same token, in order to move things forward, there does need to be some standardization. There does need to be harmonization. And so I think it does involve sort of coming together which areas should be prioritized as a broader community and ensure that those are met. But then if there are interesting or important innovations, even for an individual company that need to be made, there should still be room for that. It's true that you can't do everything, but there does need to be some sort of collective efforts to sort of figure out what we're going to prioritize as a broader community.
Thanks, Nicole. Anyone else like to build on that point?
Well, I agree with the point of balancing the innovation with progress. Sometimes we see high tech and high level of innovations applied to even biomarker discovery applications. Those might not be as applicable in the context of a clinical trial. And so there is a sudden drop of the value of creation of so many good evidence that then would not allow the actual adoption of the biomarker. So maybe focusing on tools and methodologies that are simpler but more applicable and repeatable across a larger community might be the goal when we try to achieve and pursue these biomarkers at a large scale rather than applying for a grant that might require an innovative point. And so funding and incentives that are prioritizing innovation are not always leading towards the goal of adopting that tool or applying that tool at a large scale. And I think working through the balance between innovation and progress is what would be great.
Thanks, Ramona. Any other thoughts here?
That strategic planning early on is important. It's easy to say, don't boil the ocean. That doesn't apply well to biomarker science by far and away. But you have to make sure that you're able to be adaptable, flexible, and nimble. I think of some of the programs through the last 10 years, we've gone from bulk sequencing through DropSeq to now Xenium and spatial transcriptome. And within the project, that has transformed and that technology has advanced, and it advances at such a rate. Same thing now with AI. The next day it's a different, and how to do that. But we needed cells, and we needed standardization of methodologies to get into an organ, to get enough cells to do any analysis. We needed to think that we might need saliva, we might need blood, we might need other things think of even the AD story of, we started looking much more in imaging, and what was ADNI was founded on that became CSF. That was far too invasive. And then we tested, and now look at where we are with blood and the approval of not only some really far advancing testing, but also, the non-invasive of being able to follow that disease, perhaps with blood and new approvals of drugs. And now we're on the verge of some other tau treatment drugs. I would tell you that was not visioned in the beginning, but what was visioned was a structure that supported that capability and that innovation. The types of samples we might need in the future, a repository at the same time, thinking about what may be coming. I think that strategy must be part of it, as well as the regulatory plan in alignment with FDA needs to be upfront, not something to think about when you start getting good data and let's go see what FDA thinks, but have them involved in that design, engage the review level. Don't blame the leadership. Work with the review people, develop those relationships, really have them get and understand your thinking, let them contribute as both scientist and as a regulator is really the approach that we often take. And I think that's beneficial.
Thanks so much, Steve. I wanted to see if there's any other thoughts wanted to build. Okay, so turning to the room one more time to see if there were any in-person questions. Wouldn't be sent to my iPad. Okay, so one Slido question that came in, Nicole, if I could put you on the spot because it was directly for you. So looking back on the MRD endpoint case study, is there anything you would have approached differently based on what you know today?
Yeah, there's probably a lot, right? As was mentioned earlier, I think it really is good to sort of look retrospectively at the examples and the scenarios, examples that we do have to sort of inform future learnings. And I think in the myeloma space, one of the things that's quite, obvious when you look at it, it's just that it was actually a really long process, right? And I think a lot of that time was spent in consensus building, dealing with many of the questions and topics that we talked about just during this session. How can data sharing be done? Reaching those agreements that would allow for that. I think there was a recognized need within the community, but sort of translating that into practical applications of allowing data sharing or providing, information was just an absolute challenge, right? And so I think sort of if there is perhaps that recognition of the need earlier, then perhaps there can be mechanisms through neutral parties or other private-public partnerships that could allow for sort of easing those transitions for data sharing and collation of data at an earlier phase. I do think that the standardization was really key, and that kind of came early, relatively, in the process. But I know looking at a lot of fields and a lot of within oncology in particular, there isn't that standardization. And so many of the lessons and some of the weak points that were from the MRD example, I worry we may still repeat some of those scenarios. So I think those are probably the largest areas. Is one there? I think there needs to be alignment early on about the need and the focus for within a disease space for biomarker development. It requires collaboration, and that kind of needs to happen earlier in the process, and then standardization. And so ways to sort of smooth those areas would definitely help for future biomarkers.
Thanks so much, Nicole. And as we wind down our session, I wanted to give each of you a chance to respond to the last question that we have here on screen. So what concrete actions should begin in the next 12 months so that within 5 years, multiple disease areas have active collaborations generating reusable biomarker evidence? And to embellish a bit from what's on here, I think we're really trying to drill down into specifics here, whether it's 12 months, 5 years, 3 years, but really curious about what are those near-term steps that can have those sort of near midterm effects within generating reusable biomarker evidence. So I feel like we can just go down the line here. I'm like, will there be Ramona or Steve who'll like to start us off? I don't, everyone's laughing. Let's see. Ramona, do you mind just starting us off as we wrap up the session here?
Sure, and of course it's high bar for assessing what should be done in the near term, but I think what the process of aligning on what's not working, I think is the most relevant now and the one that we can assess and potentially work through in the near term in terms of understanding the unmet needs as a community, and then perhaps create some pilots for disease families to work towards specific common areas that might be downstream enough so that the shared investment incentives are larger. An example might be looking at the neurological, neurodegenerative disease space versus the neuromuscular space, and really trying to find where the gaps are and bridging into how we can speed up the development for those areas that really do not have high numbers or or such non-natural history, so that those that do have that historical background can support communities and unmet needs for the future. So the notion of reusable evidence that comes from one disease area to the other might be early focus for the near term, as well as, again, working through the enforcement of those notion of how we can incentivize sponsors to make sure that there is an opportunity to use this data for the future, and perhaps create solutions where we request everyone to have a data management and sharing plan ahead as part of the protocol, so that we can then structure and predict what the future will look like for sharing the samples downstream, even in case there is a potential drop of the program and closure in a termination of the program early time. So being ready and understanding that any single data we collect and any single specimen we collect will be relevant is what we think, I think we should be start working on in the near term.
Thanks so much, Darcy.
Great. And I love concrete action questions. That's awesome. I definitely want to see more of these types of workshops like the limb-girdle muscular dystrophy or the MPS workshop that focused in on biomarkers. I think a near-term goal would be having these third-party conveners really identify the promising biomarkers that can focus where we can best have this early alignment, and it just gives something that's a little bit more achievable. I also feel like I'd be remiss if I didn't mention RISE workshops, because I feel like that could be a leverage, like it could be a lever in order to do this type of discussion. It just depends if that's something that FDA is also interested in pursuing. But overall, just want to have results that yield sort of a disease by disease map of where biomarker evidence is actually feasible, and moving forward from there.
Thanks so much, Darcy, and appreciate the plug for our RISE workshops, which are a series of rare disease workshops that we co-convene here at Duke Margolis with FDA. So we have another one coming up in September, so appreciate the nod there. Nicole, and then Steve, you'll take us home.
Yeah, I think I would just echo a little bit of what I said before in terms of just really underscoring the importance of harmonization. But I would point out that it's important for us, I think, as a field and within specific disease areas to sort of align on where there are opportunities to sort of coalesce around efforts, right? When thinking about certain biomarkers, is it appropriate, to just, for example, how can we pool data even and sort of advance standardization or harmonization across various diseases if there's a similar biomarker, like ctDNA being an example. And then also, with it across disease spaces, are there areas where there may be learnings already, but we could extrapolate them to other settings, and what information do we need to sort of extend? So really questions around where is it appropriate to lump together data, have shared efforts, and then where is it not appropriate, all with a goal towards standardization and harmonization so that there ultimately is a better data package down the line.
With all everyone had said, it's easy to not have any time left and go last. Can say everything they said we should be doing, because I agree. I read it a little bit even differently, and I don't want to, by any stretch, leave out somebody's disease area or something's more important than others. But I do, without running toward the shiny penny, I guess there's no penny, shiny nickel, that rare diseases, and how can we extrapolate data from one similar type of rare disease for biomarkers that can drive drug repurposing, that can drive specific development of therapies for rare. We just have to make a difference. I talked about neuropsych. I mean, this is the modern problem of our world. We must go after that with robustness and effort. I think again, I don't want to go generally, but I think about cross disease, fibrotic disease, and chronic disease, whether it's in the chest, whether it's in MASH, whether it's in where that be, there are areas where we have to have harmonization, standardization, utilize parallel data sets to be able to go across disease to go after that effort. And then I could easily, I would never forget the cancer world and the idea to apply learnings from myeloma to AML or even to breast and other areas. It's not that we're not trying to do these types of things, but really a strong investment and alliance with FDA. Yes, there's 50, 60, 1,000 things we need to do, but where the unmet need is really large and really impacting us today as a people, for lack of a better way to say it, we must invest there. We must go after and try to really solve some of the great chronic burdens upon us. So maybe that's a nice way to stand it and push us with a half glass full, but half glass empty type of approach.
Well, thanks so much, Steve. And thanks again to all of our panelists for a really, really interesting discussion.
Hi, everyone. Welcome back. I'm Julie Tierney, and I'm pleased to moderate our third session, Improving Biomarker Review, Transparency, and Engagement Across Development. And now that we've discussed biomarker evidence development and the infrastructure needed to support it, we'll turn to how stakeholders can help build a clearer, shared understanding of complex and context-specific biomarker review. In particular, we'll consider opportunities to communicate review learnings more effectively, support more actionable engagement at key development milestones, and help stakeholders understand how prior regulatory experience may or may not inform related programs. I'm joined today. I'm a principal at Levitt Partners, which is a healthcare policy firm based in DC. Prior to that, I spent the bulk of my career at FDA in a variety of roles and have worked on biomarkers throughout much of that policy around the biomarker qualification program, codifying it into law, establishing it at FDA, and working on accelerated approval in the Center for Biologics and across the centers. So I'm thrilled to be a part of this discussion today. Next slide. Or do I need to do it? Sorry. Where did I put it? Yeah. Did I advance it? Okay. Oh, yeah. Great. Thank you. Sorry. Okay, so I'm thrilled to be a part of this discussion today. I'll introduce panelists briefly, allow them to do some introductory remarks. We have Jeff Allen, who's the president and CEO of Friends of Cancer Research, Allison Barrett, who's the chief science officer at the Foundation for Angelman Syndrome Therapeutics, and Rob Berlin, who's head of regulatory policy at Vertex Pharmaceuticals. So why don't we start with Jeff? Thanks for being here.
Great. Yeah, thanks for the opportunity to join the discussion. As Julie said, I'm the president and CEO of Friends of Cancer Research. We've had a longstanding interest in the biomarker space. On the scientific side, we've run a number of consortia around the harmonization of different biomarkers, efforts to validate different biomarkers and endpoints and technologies, all aimed toward optimizing drug development in the future. Alongside of some of our scientific activities, we've had the chance also to work with a number of folks on the panel and in the room today around some policy proposals that I think are coinciding with a lot of the discussion and the roadmap that the Duke team has put together here, largely around how to enhance the biomarker qualification program. Based on some research that we had done, kind of doing a landscape assessment around the experience with that particular program over the last 10 years since it was codified into law. And the long story short is a good number of biomarkers have gone into the program, but not a lot have come out, whether that be in terms of validation or withdrawn. So I think there's an opportunity, particularly given the advancements that have been made in technology, that now could be a good time to re-examine that particular component of the framework in thinking about engaging with the regulatory experts during the process of biomarker development and qualification.
Great, thank you. And I'll pass it to you now, Allison. I also wanted to take moderator's discretion and say that hopefully you'll be helpful talking about biomarkers, but also we'll have some color into intermediate clinical endpoints as well.
Yeah, thank you.
Thanks for the invitation, and I'm really happy to be here. So I'm going to reintroduce myself because I wear many hats, and I think those hats are incredibly important to the discussion. So firstly, I'm a veterinarian by training. I'm an internist and an interventional radiologist working in medical device development, hepatobiliary diseases, and renal diseases. So I understand biomarkers and endpoints for peripheral pathologic disease very, very well, and I run clinical trials in those areas for my entire career. But I also serve as the Chief Science Officer at the Foundation for Angelman Syndrome Therapeutics, which is a therapeutic developing foundation really focused on advancing cell and gene therapies and disease-modifying therapies for a rare neurodevelopmental disorder called Angelman syndrome. So I really appreciate the panel before because I think we really brought in and highlighted the major challenges, and thank you for your remarks about rare disease, because I think rare disease is a totally untouched and unmet area that is almost like an elephant in the room when it comes to biomarkers. And so hopefully I can really represent the challenges that we face as it relates to biomarkers. I'm the co-director of a pre-competitive consortium, which also highlights kind of the last discussion, where we actually have 34 industry members, 350 members in total, patient advocacy groups, clinicians, researchers, and we create this pre-competitive space over the last 10 years, having spent over $5 million developing clinical endpoints and biomarkers. We've developed many clinical endpoints, a master protocol for clinical trial design. We have 3 phase 3 ASO studies, one of which just completed, and an AAV gene therapy in the clinic and a CRISPR on its way and an HSC autologous gene therapy right behind it. But what I would say is that after $5 million, we still don't have a biomarker. It's not for the fact that we haven't tried. It's not for the fact that we haven't unturned every single stone. It's for the fact that this is a neurodevelopmental disorder, severely debilitating neurodevelopmental disorder of neurons, of the brain. There's nothing peripheral that's abnormal. There's nothing anatomical that's abnormal. Imaging is normal. But unless we do a brain biopsy, it's very hard to find a fluid biomarker, whether that's in CSF or whether that's in the periphery. So that leads us to say, maybe we'll find a signature of proteomics, maybe we'll find transcriptomics, maybe we'll find a protein that shouldn't be secreted in the CSF that might be there. But the amount of untoward burden to families and patients who are nonverbal, who need to be anesthetized for every lumbar puncture in order to try to screen their CSF time and time again to see if we can get more and more sensitive to find something that might actually allow us to have a surrogate endpoint is really challenging, and it's a real tall ask. And we are one of the more common neurodevelopmental disorders. We have 500,000 people in the world potentially with Angelman syndrome. What about most of the rare diseases, rare pediatric diseases? 70% of them are neurologic of the pediatric population, of which 50 to 100 or 500 people exist with that disease. What are we doing from a biomarker perspective? We have to be able to share all of this data for us to understand how we actually look at biomarker endpoints and clinical endpoints. And in these phase 3 clinical trials for an ASO for Angelman syndrome, 3 different companies, we have children completely nonverbal talking, but we don't have a biomarker. But that is not enough for an intermediate clinical endpoint without a randomized sham or placebo-controlled study to show how it compares to the natural history of the disease. Well, we have 600 patients in a 20-year natural history study. But that's not enough because that doesn't accommodate the placebo effect that might be had with these genetic therapies. And so you're really stuck in the middle where we're trying to find biomarkers. Our foundation has spent $5 million, but we haven't found one yet. But we have an EEG that's pathognomonic for the disease that improves with a delta spectral power over time, which has been associated with cognition and IQ and activities of daily living and neurodevelopment. But EEG has not been validated as a biomarker. So what do you do when you have change in EEG that's supporting target engagement? You have children that had no words that are speaking, that are sleeping through the night for the first time in their lives, and that are self-feeding, having never done that before. And you don't have a fluid biomarker, where you're left with saying the end of phase 3 will tell you if you get an approval, but there's no path for an accelerated approval for these populations. And I think that's a really important point for neurologic disease, and most rare diseases are neurologic, and most of them are not degenerative. So we're not having NfLs, we're not having heparan sulfate, we're not having these enzymes that we can measure in the CSF. So I think it's really important as we come together to think about what is the path for those that don't have a biomarker, or
let's think about the biology of the disease and how we could be adaptive for fit for purpose, where maybe EEG for neurologic disease is the right path. And let's come up with a way to help support that for these neurologic disorders rather than dismissing it, which is what's been happening over the last few years. So I think it's really important we come to terms with digital health technologies, EEGs, and clinical endpoints that might actually be intermediate, that can help tell us how we can look at the future for these populations and meet the needs of patients, get them treatment, and then follow through with what does the evidence look like over time. And I think that's the way we really treat these rare diseases.
Thank you very
Great.
much, Allison.
Yeah. It's like, welcome, Rob. Yep.
Thank you very much. Yeah, so Rob Berlin, I'm from Vertex Pharmaceuticals. Yeah, there's a lot in what you're describing. I think when we think about rare disease, that's kind of top of mind for me. Biomarkers, really, we think about them in all of our development programs as we're trying to get to patients sooner, get therapies out there. And I think, a clear theme to me is this sort of uncertainty that we look at. We're looking at biomarkers and we're trying to aggregate across different areas. So, when I think about your example, you think, well, what can we learn from a variety of neurodevelopmental diseases, and think about what can we borrow, what sort of examples can we carry between them. And so a lot for me is an interest in kind of bridging and how we think about carrying from one context to another. And as part of that, I think it's getting to some interim wins. So it's getting to things that are, having clarity on where a development program isn't quite meeting FDA's expectations, for example, so that we can figure out how to bridge those as quickly as possible and as early as possible in our development programs. Another is thinking about, can you get labeling statements? So even if something doesn't end up being, a definitive marker that's your primary endpoint. I took Isam's point very well about secondary endpoints and exploratory endpoints, but to some extent we are looking at those issues in part because the agencies are telling us to look at those issues. And then you need to figure out, are there ways to get those into labeling so that you can sort of get those wins on the way, and you can communicate back to patients the value that you're seeing, right? That in fact your experience and your description of what your challenges are is actually being reflected in what's approved. And I think part of that, there's an idea of really, I understand there's going to be variation between different review divisions, but having as much commonality to the structures of the way that we're presenting data and what we're getting back from the agency would really be beneficial so that we can understand the gaps. Because I think currently one of the real challenges when you're working with a regulator is that we have people who are true experts in their disease area, right? And as experts in their disease area, part of why they're in their positions as office directors and division directors is because they've kind of internalized a lot of this experience. They understand like there's unmet need and there's really, really unmet need, right? And there's sort of, those things can be hard to pick up as a company. I think even harder for patients and other people outside of the agency. So getting to a place where we have more regular information, both in terms of what we're sending in and what we're getting back, would be very helpful. So it's a great discussion and really look forward to it.
Great. Thanks to all of you. Next slide, please. Great. So as we begin our discussion, I'd like to point your attention to the Slido poll up there on the screen, which you can join through the QR code or by entering the code. And we'll leave it up for a few minutes to give everyone a chance to access it. And what we'd like to do for this polling exercise is we've listed the policy goals in 2 and 3 and ask folks to please select 3 that you think would be worth further discussion or consideration, respectively. And I can read through them as you consider them. Number 17, expand public case examples, review summaries that show how biomarker evidence was evaluated in individual submissions, including accelerated approval cases where surrogate endpoint evidence supported approval and later confirmatory trials did or did not. Verify clinical benefit. 18, clarify how prior regulatory acceptance or use of a biomarker may inform later product-specific review, including how FDA weighs the degree of regulatory novelty when a biomarker is proposed in a new context. 19, identify recurring evidentiary issues that limit biomarker use and review, such as biological plausibility, clinical relevance, longitudinal evidence, the biomarker change and clinical benefit relationship, analytical validation, and assay comparability. Number 20, develop clear public expectations of how FDA assesses the clinical meaningfulness of biomarker changes in different disease contexts. 21, strengthen internal coordination across review divisions, offices, and centers to support more consistent interpretation of biomarker evidence. When individual product submissions raise cross-disciplinary considerations. Next, increase visibility into novel surrogate endpoint review process for sponsors. 23, maintain specialized biomarker, translational science, statistical, clinical, and assay measurement expertise needed to evaluate complex biomarker evidence and conduct subject level and cross-program analyses when appropriate. Number 24, clearly distinguish when review concerns relate to the biomarker suitability for its intended regulatory use versus the size, durability, or clinical meaningfulness of the observed treatment effect on the biomarker. Next one is clarify validation expectations for biomarkers with prior regulatory use or substantial development experience. And then next one, via public-private partnerships, identify therapeutic areas where biomarker policy interventions may be most useful and publicly explain why those areas are appropriate. Next one, provide structured biomarker-related feedback, especially during pre-IND, end of phase 2 type C and other relevant meetings before sponsors commit to late-stage trial design, endpoint selection or accelerated approval strategy, and then create and pilot an earlier structured cross-functional engagement opportunity focused on biomarkers. Clarify which meeting types are best suited for key biomarker questions, like exploratory biomarker use, endpoint selection, surrogate endpoint strategy, et cetera. 30, identify the development milestones where biomarker feedback is most consequential and proposed structured engagement there. Support multi-party engagement among sponsors, assay or diagnostic developers, and relevant review staff. Include relevant disease area reviewers and centralized biomarker, translational science, and statistical experts in early biomarker strategy discussions. 33, establish clearer mechanisms for incorporating patient input into biomarker strategy discussions. 34, FDA sponsors and neutral conveners should pilot disease-specific endpoint development discussions in areas where recurring disagreement about biomarker endpoint selection has delayed late-stage development. I think that's all of that. So if you could select 3 from goal 2 and then 3 from goal 3 that all of which warrant further discussion. Great. So we'll give folks a minute or two. All right. All right, I think, well, let's see. I think we're about done. Yeah. Okay, I think for goal number 2, looks like the top 3 recommendations that warrant further discussion are identify recurring evidentiary issues that limit biomarker use and review. Then, no, sorry, let's see. I guess that jumped to number 3. I don't know. Is Patrick handling the poll? Okay, well, we'll just go to these top 3. So we've got the recurring evidentiary issues topic, and then the providing structured biomarker related feedback early on before sponsors commit to late stage trial design, and developing clear public explanations of how FDA assesses the clinical meaningfulness of biomarker changes. We could just scroll up one more, and then expand on public case examples which I think probably goes well with number 20. And then finally clarifying how prior regulatory acceptance of biomarker use of biomarker may inform product-specific review. Sorry. All right, but now I think we can turn to sort of looking at those. If we can go to, I think, the next slide with the discussion questions for the panel. With that in mind, and then I'd like to go down the panel as we from Jeff, Allison, and then Rob, and ask, which goal 2 or goal 3 recommendations seem most useful to you, and which seem least feasible as currently framed. I'll start with Jeff.
Sure. I guess I didn't vote because I was sitting up here, so I don't know if this would have swayed it, but I'll comment on the ones that came in around I think maybe 4 and 5, number 17 and 18, about ways to communicate past experiences and prior regulatory acceptance. And the reason that I think that those are important ones, I'm glad to see that they made it in the top 5. As I mentioned, working with a group of stakeholders, one of the things that this particular group of experts that had experience in developing biomarkers and regulatory expertise thought would be helpful from a regulatory standpoint toward qualifying biomarkers was to have an opportunity to recognize that the information about a particular biomarker matures over time, and how to recognize that evidentiary journey as new data is emerging. And so what was proposed was more of a sort of tiered framework than what is available now through the current biomarker qualification program. So to be able to recognize that, when a tool is at a initial tier, where there may be some analytical data around the use of a tool to measure a specific biomarker, and then a subsequent tier that would be able to identify biomarkers that would get to the issues around biological plausibility and a high degree of interest in terms of developing the data that would be needed to advance it further. And then a further tier around recognizing the prior use of the biomarker, when it has been incorporated into clinical studies, when it's been used for supporting evidence, perhaps it's been used for an accelerated approval previously for a specific context of use. And then that final being sort of the full qualification. And one of the reasons that we discussed having this sort of multi-tiered type of approach is to also be able to recognize that there's different pathways that biomarkers are going through their development of evidence. One is a formal qualification program, but the majority of them are more in the individual IND-based development program. And so how can you bring those programs a little bit closer together to be able to leverage regulatory precedents, which is an important component of this. So there may be biomarkers that exist at tier 3 if they've been used as an endpoint in a particular context of use, and that may be fine for them to stay there, for eternity if there's no other potential context of use that could be explored. But then, based on the current paradigm, one has to sort of go back to the beginning and start over if you want to identify another context of use where this tiered type of structure might be more conducive to not having to start from scratch if there were the evidence to support an expansion of a context of use. So all that to say, are there better ways to communicate the use of a biomarker more transparently and more regularly? And, a lot of this information is out there. It's just sort of trying to put it all in one place alongside what the regulatory acceptability of that particular biomarker in its context of use was. So one opportunity could be, trying to leverage things like the summary basis of approval. And while those are available documents, the ones that are most available are those that are associated with the initial approval. And that may not be the space where a biomarker has been incorporated into a study. So are there ways to leverage these types of documents, make supplemental basis of approvals more readily available in a more timely fashion to be able to recognize when a biomarker has been used for a certain context, whether that be the basis of the approval itself or perhaps in dose determination or other considerations around characterizing that therapy. I think we're seeing an interest in a greater level of transparency for some of these review documents. The agency has taken initiatives to make complete response letters more available. And maybe that's one other venue for perhaps if a drug itself was not sufficient for approval, that doesn't mean the biomarker didn't provide some valuable information. So are there ways that these review documents that could be leveraged more regularly? I think would be a very valuable way of trying to align clarity around the use of biomarkers. And I know that may cause a little bit of angst in thinking about, well, there may be proprietary information or, trade information that we may not want to be disclosed as readily. But I would say around the biomarker and the application and its development program, a lot of this information has probably been presented at scientific conferences before. And so, of course, for these documents, there should be the right redaction. But I don't think that the biomarker utilization is necessarily a proprietary piece of information, and for the good of the field and for the people who ultimately will benefit from the next generation of therapies, it probably shouldn't be proprietary information to one single developer.
Great, thank you. So Allison, I'll turn to you, and I think let's do the first two questions. So Jeff did that, and I think that'll helpful.
That sounds good. And I think really what I highlighted as important to me fit into the kind of first two things that were in the priorities, I think, that the group in the room said. But ultimately, my feeling is, for the question of what kind of public-facing learnings and case examples, like explanatory materials, would help sponsors and stakeholders. One thing I think is important is we think about all stakeholders, right? So it's important we target all stakeholders. And two hats I wear that I didn't actually mention is that our foundation started 4 companies in order to develop cell and gene therapies. So I'm wearing an industry hat as a founder of multiple companies, co-founder. And I'm also wearing the hat of a mother of a little girl who lives with Angelman syndrome. So I'm her mother. And so we had to develop these drugs for her and the other 400,000 kids like her. And so when I think about stakeholders at the table, I think it's really important that we think about the learnings we have from burden of disease. Being one thing that I really liked was the idea of piloting early engagement. And that's not only early engagement with a sponsor, that's early engagement with the advocacy groups, that's early engagement with patients and families, that's early engagement with regulators, helping to drive and talk about what's a reasonable biomarker. We have spent so much money to come to realize that may never be accepted, but there's not really, everyone always says come early and often. In what forum, right? When you exhaust your externally led patient-focused drug development meeting, when you exhaust your patient-focused listening session and your patient advocacy group, there's really not a forum to be able to come early and often. And so having an opportunity to just meet, to say, this is what we're looking at from a consortium level, bringing sponsors, bringing parents, bringing families, and say, this is what we're thinking. Do you agree with these criteria? Yes, we do or don't. Come back to us when you've got more information. And then we have driving orders in order to not just spend millions of dollars up front, but actually think about where we can have go and no-go decisions along the way. But instead, to your point, what happens is that it gets put into somebody's IND because it's a faster path to feedback. But whoever IND it's in, because it's usually not the patient advocacy group's IND, unlike us, you don't get that feedback as a patient advocacy group. So you keep spending because the sponsor keeps telling you that there's a path here. It's looking good. We're getting good feedback, but we don't necessarily see the feedback until they're at their end of phase 2 meeting or starting phase 3. And they're like, they're never going to accept EEG, sorry. And it's like, well, we just spent over $1 million on trying to validate EEG to cognition. And we're told now they're never going to accept that. So it's important, I think, that you have an opportunity to meet often. I think an opportunity to meet with all stakeholders and really think about the patient burden in what we're requesting and asking for, especially in rare disease. So if we're looking at a CSF biomarker, we have to think about they want samples day, night, serially, monthly, seasonally, because it can change. And there's rhythms of which proteins change in the CSF potentially. Well, if you have to anesthetize a patient every time you get that and you want 4 baseline samples over the course of a year, the burden to those families and those children and the risk with no benefit is real. And so if we're not having those conversations with the agency to explain what it is that really is hard and how that's functionally difficult, and it's also financially close to impossible. Lemonade stands, golf tournaments, and cupcake sales can't pay for this. How do we have a path forward? What can we do to think about that disease in that situation? And I'm speaking on behalf of all rare. We're all dealing with the same thing. So if we just say, let's come up with a plan and a pilot for EEG for neurodevelopmental disorders, and let's try to see a path so that we can all come together as multiple disease organizations and pay for that path. Share the data we learn, share the feedback from the agency and that the sponsors are getting as it relates to those types of biomarkers. And then we can actually advance the field. And so for me, I think public-facing learnings cannot be stored in an IND. They have to be shared, particularly in rare disease, because the only way we're going to actually be able to advance the field and to be able to look at adjacencies of similar diseases is to be able to share those learnings and have visibility into the feedback that people are getting. And biomarker development is not easy, to your point, but it's even harder for rare disease of the brain. And so we have to think about that in a really, really different way.
And it sounds like not waiting until you can't wait until
Yeah, wait till
there's
there's
decision approval.
Wait till end of phase 2, because for rare disease, you're exploring this in phase 1, right? People aren't going to just get a CSF tap for no reason before a phase 1 starts. So you're going to be doing a phase 1 looking for target engagement, doing serial CSF taps to try to discover the biomarker in the phase 1/2. Then you're going to bring that to the agency at the end of phase 1/2, but it's not nearly validated. So now you have to try to validate during phase 3. It makes accelerated approval very, very hard, if not impossible, if we don't come up with an easier path to think about these rare indications.
Thank you. Rob, what did you thoughts on this?
Sure. Yeah. So I think I was thinking a lot about that, sort of how you get information out on the FDA's rationale for why a certain biomarker is not going to be picked up, right? And I think it's a very fair point that one of the limitations is from industry and what's disclosed and what's shared. And it's tricky because I think in a lot of cases there's information that's not shared because the company's intending to come back, and that adds a bit of a challenge there, right? Is that there's a desire, and this is the agency's constraint, right? Is that they're looking at it and saying, well, we can't really disclose this because the company hasn't, even if it's a CRL, the company hasn't abandoned the product. They haven't succeeded on that first cut. So that's a very, very tricky question. I do think having kind of standardized ways of submitting the information and having standardized ways of getting feedback can help with a couple things. One, it can make it very clear what the basis was for the agency's determination, which I think is important. So if I do think one challenge is that since it's kind of an aggregate of totality of evidence kind of analysis, as a company, you're sometimes in a spot of saying, I'm not exactly sure that I could tell exactly what the reason is for this rejection at this point, right? So you have kind of a limitation in what you're able to share based on your own understanding of what you're seeing in the feedback. So I think the standardization would help with. I think it would also make it much easier to aggregate outcomes across different products because you have a common way of describing it, which I think would help with disclosure from the agency side. If they're saying there was a section that was about population size, the population size failed to satisfy because you had X issue, like you're going to need to amplify that because you haven't shown us enough on these other areas. So it could start to give some structured way to laying out that information. So I think that is part of the process when we think about how we get the information out. And I do think for companies, there is a desire though to build those relationships with patient groups. So I think having some of that structure would help too, because I think there is generally a want to go back to patients and explain what's happening. You also run into questions though there of like, you don't want to be acting improperly in how you're doing that, right? Like you don't want it to be, well, we got denied on this endpoint, so we went to our patient group and explained how FDA took sort of an irrational position, because then you're in sort of a weird spot, right? And I'm not saying they are taking an irrational position, but that can be the perception, right? Of you trying to be candid, yet does it look like you're actually just trying to sort of game the system, right? So I think it's a really tricky question. I think the more that we have common ways of describing things, the easier it is to aggregate, and the easier it is for us to sort of concretely say this is where the challenge is, as opposed to it being sort of like, understandably kind of a gestalt feeling about like, this isn't quite ready yet. And it can be not quite ready yet because the magnitude of the impact wasn't what we wanted, or it can be not ready yet because we think there's such a small population that we may be picking up just some change that could have happened absent the therapy altogether, right? There's placebo effects, those sorts of things. So I think those sorts of creation of structures that are common can help with that aggregation. And I think then too, it'll help us as we try to aggregate across a variety of areas, right? If you're saying these are all a set of diseases that are impacting, the same organ, and we're seeing similar outcomes across them, we could reasonably guess that like, that's, I don't know, if you're reducing FEV1, that's probably an impact on lungs, and all of these diseases may be doing it in a different way, but it's probably a good measure, right? And you can borrow across them. And I recognize like the neurology is a great example because it's so much more complicated. The lung volume can have challenges in younger patients and those sorts of things, but it's an easier objective measure than some of these things that are truly variable across different disorders and within heterogeneous populations in the different areas. So I think there's a lot there. It's just sort of trying to figure out all the structures around how you do it most effectively.
And I think that's where being able to meet often, to be able to say, where are we starting from, right? If there's a reason EEG from the get-go is not agreeable, why? But let us know, because we shouldn't spend $1 million on developing EEGs and correlating that with natural history of disease if it's not acceptable.
It's never going to be.
And if it's never going to be acceptable, or these are the reasons this is what we would need to see to even consider acceptability. And even like CSF biomarkers, which seem great, right? Like in the CSF, you can see a protein signature potentially for a disease, except one drop of blood, you now have protein in the CSF you're measuring that's not from the brain. So now what do? So now you need to have a plex model where you can measure hemoglobin and white blood cell metalloproteinase in order to confirm your sample doesn't have any white or red cell contamination, to know that the proteins you're measuring are coming from neurons. Why did it take us 4 years to figure that out? If we had an opportunity to meet about, we're developing this type of biomarker, what are the trials and tribulations of doing that, and what would you need to see if there's precedent in another disease in which they found this challenge, and therefore we're going to come back to you and tell you this is, I would add this right away from the start if you're going to start looking at a protein signature, because you're going to get stuck with a lot of blood contamination. And it's true, a needle in the spinal fluid, you're going to get a blood contamination. And you have to minimize that and figure out what is your path, if that's a path at all. So even if it's those early things, not like what does your evidence show whether it is or isn't a biomarker, but even like, is this worth developing?
Well, and I'd just add to your early conversation. It's early conversation with all the right people in the room.
With all the right people in the room.
I think it's really like
it's fair and balanced. Agree.
Well, and I think from even from the regulator side, right, is you have your clinical experts, but you have the people who are going to ask the questions about your modeling of your pharmacodynamics, like having all those questions asked at the start, is really helpful, especially when you're in kind of the uncharted territory of a rare disease.
Yeah, totally agree.
Yeah, and I think it's something that I've heard in a lot of these conversations is, these early collaborative discussions and it being about what the path forward is.
Yeah.
Not a no, but knowing all of the known unknowns, is there a way to advance and what's realistic, right? So I wanted to return back to the poll, if that's okay, just definitely seeing recurring themes under goal 2. I just wanted to circle back that the top 2 goals that were identified were identify recurring evidentiary issues that limit biomarker use and review, and then develop clearer public explanations of how FDA assesses the clinical meaningfulness of biomarker changes. So I think that really resonates with the remarks that you all have made so far. And then under goal 3, the top 2, we're providing a structured biomarker related feedback before sponsors commit to late stage trial design, endpoint selection, or accelerated approval strategy. Certainly getting advice that it's going to be a review issue may be a little too late, I think. And then pilot disease-specific endpoint development discussion in areas where recurring disagreement about biomarker endpoint selection has delayed late-stage development. And I think certainly with some of the neurodegenerative diseases, we've seen that. So I will return back to the next set of there's 2 more questions, I think, for discussion. And I think, thinking something that Allison, that you brought up, but, are there biomarker strategy questions that can be addressed through existing FDA meeting types, or, where are there more structured or cross-functional engagement mechanisms that could be useful? if it's okay, I'll start with you and then Rob and then Jeff.
Sure, sure. And I think Rob probably can do a better job than me as a regulatory expert here, but in terms of what are the options for all the meetings. But what I can say is that generally from a patient advocacy perspective and from a small biotech startup perspective, you may have an interact meeting, which this is way too far advanced for an interact conversation. And then you have a pre-IND meeting in which you have no data to support anything. This is what I'm thinking about doing, but it's generally not related to biomarkers. You're just making sure you got your GLP tox right. And then the next time you actually meet is your end of phase 2. Now you can have type C meetings in between, and if you're a patient advocacy group, of course, maybe you had a patient-focused listening session early on, but that you have one of those. And so that happened for us in 2018. So that's done. Our externally led patient-focused drug development meeting was 2025, which is about clinical meaningfulness and what's important and clinically for families and caregivers. So that's done. And so now you're in a point in which you really have to almost wait for your end of phase 2 or think about a different strategy to just talk about biomarkers. And it may be a very brief meeting of this is what we're thinking. What do you think? And can you give us feedback? Of course, you want to come in with a position of information you have, but a lot of it is not going to be post-treatment data. It's going to be pre-treatment data, baseline data, natural history data to say, is this worth further pursuing? And that's an open-ended question, which is a hard thing for anyone to give you feedback on. But that's where you start. And if you don't start there, if there's a very clear no, because we've done this before, and just so you're aware, it doesn't work for XYZ reasons, then you're going to go down a path to try to get some evidence to see if you can get feedback on that evidence. But that process can cost millions of dollars. And in rare disease, you don't have that, especially in these small startup biotechs where that is a very hard thing to come by. So for me, I think it would be nice if there are these brief meetings you can actually have, not written response, but a conversation with all the right people at the table to give you clinical feedback and biochemical feedback and really understand what the nature is of what you're talking about. And bringing clinicians and families to the table to say, I know you would like 4 CSF taps in a year, but that's not feasible. We can't do that. And these are the reasons why. Therefore, what are the alternatives for adaptability to the clinical disease we're talking about?
Thank you.
Yeah, I think it's actually, I like your framing of it quite a bit. I mean, I think one of the, this is an idea I take from someone else, frankly, but there are the surrogate endpoint meetings. So there are opportunities to go in and have those direct conversations about surrogate endpoints. And I think that one of the big benefits is you're tied into PDUFA timelines and commitments, which I think is really important. I think when you're having a meeting, we talked about things like patient-focused drug development meetings, right? If you're having a patient-focused drug development meeting, one of the challenges is you're not sort of looking at timelines and concrete outputs, right? So I think you do benefit a lot from being in the structures that they have around things like a surrogate endpoint meeting. I do think Interact has some potential, as long as you're hitting it at the right point. And then maybe there's more opportunity to use some of the type D meetings to have some of these conversations to say, like, there's 1 or 2 discrete issues that we want to address, but it is challenging on sort of the other side that then you've like, well, I've got everything baked except for this 1 or 2 questions.
Yeah.
But then, how likely is that when you're considering your early trial design, right? So,
Yeah.
I mean, I think I would look a lot at the surrogate endpoint meetings, and then in general, just saying, having some way to signal to the agency how sort of pivotal this question is within your application, so then you get the right people at the table. I mean, you can request to have people at the meetings, right? You can request to have certain FDA staff, and I think, that's something that's definitely worth thinking about in these spaces when you have it maybe there's probably a tension between like having early and you're not going to get early, often, and comprehensive, right? You're not going to get early, often, and everyone from the agency at the table. So maybe it's thinking about how we do that best to say, like, this is like, it's not a type A, but it's almost a type A meeting in the sense of like, this is going to be pivotal for whether our program is going to proceed in any meaningful way. I mean, I think your example, I'm sure, from the agency side of kind of going through this process and then finding out that the EEG is never going to work is not what the agency wants either, right?
Yeah.
Like that's not a desirable outcome to anyone. So, yeah, I think that would be my sort of best answer to that.
I think probably one thing worth noting, a lot of the existing meeting types and mechanisms for engaging FDA are probably more relevant for the initial discovery and characterization of a biomarker in the context of an IND. for all the discussion today around the role that consortia are playing in terms of advancing toward qualification or broadening the application of a biomarker, many of those meetings don't exist. So oftentimes we rely on, the expertise, but also the time charity of the review divisions, who are willing to engage on some of these discussions from the consortia standpoint, because we don't have an IND. It's more of a scientific exercise and an attempt to aggregate data from different sources together to generate the necessary evidence that would be required to move toward qualification. And while oftentimes, at least in our experience, the review divisions have been generous with their time and providing their advice and expertise, but those things aren't also sort of on the same schedules. They're not PDUFA supported meeting types. The feedback may not be quite as transparent, and they have to sort of mix this with their user fee daily activities, which becomes a challenge and an onerous burden on the review staff in order to receive that type of engagement. So I think it depends on sort of where the biomarker is and what the plan is, I suppose, in terms of what the availability for engaging FDA are. And for most of our consortia, maybe FNIH and Critical Path Institute have a different experience, but having never managed an IND, I'm not sure how we would go through some of those processes of engaging from sort of that standpoint, and trying to draw the equivalence of when to request those meetings, how to structure them, and things like that. So it is sort of a different ballgame.
Yeah, and I think you really raise a good point, Jeff, that, the sort of IND type meeting and NDA BLA meeting types versus relying on sort of the time charity of the review divisions. And I think, there is a balance, because if you meet with everyone, then you can meet with no one. We really, when I was in CBER, tried to do our best to do as many meetings as possible, which required a lot of early days, late nights, but but then balanced again against sort of structured meeting types that do give some prioritization to things. And so that's tough. I think, FDA really works hard to do, patient-focused drug development, patient listening, but need to move to engagement.
Yeah, and we've been very lucky. We've had amazing engagement with the FDA at our consortium level, mostly on clinical endpoints, not on biomarkers, but we've had an amazing experience with the Rare Disease Division and COA for Angelman syndrome. So I have to say they've been great, but also that advice is generally not binding. And it's generally a bit vague. It has to be, because they don't know, they don't have a briefing document that they're going in after, right? They're going in after open questions and conversation and dialogue. But it would be nice if there were more brainstorming sessions, even in small groups, not in a consortium setting where there's 1,000 people in the room, but like small groups where you could bring in all the stakeholders and have those opportunities where we come to them rather than them having to come to us, making it easier on them because they do come to us a lot and they're wonderful about that.
Yeah, so I see Amanda was raising her hand, but yeah, I was going to say, why don't we open it up for questions from the audience? And Amanda, you go first.
That's on. Okay. Hi. This was really great dialogue, and Allison, brilliant. Everything you were saying, I was nodding right along. One area that I'd love to hear more from the group is that engagement between patient community and industry, and specifically having worked so closely and continue to work with patient advocacy groups. One thing that just resonates with me and that I've heard is I'm so grateful that there's a company that's developing in my rare disease. I don't want to ask for anything. I'm just grateful that they're developing in it. And there's a part of, yes, you can be grateful, but also, and this is the part of, if the trial fails, what happens to that data? And many times it gets put away and never to see the light of day again. And I've, on the flip side, as someone who's worked for rare disease consortia and try to get failed data, it's near impossible. And so there's a point of, I'd love to hear if there's dialogue or considerations on informed consent, and expectations of, you know, before a trial even starts, as part of the informed consent, what happens to the data if the trial is successful or fails, and an opportunity to have it in a centralized type of repository across, rare disease, something like that. That way it helps other diseases while also, really puts it back on, patient focus. Because when patients say to me, and a trial fails, it feels like it's my fault. Like that's really is hard. And then you can't get that data. Like you're saying, the cerebral spinal fluid, the TAPS, all those things. And then for that data to just go and disappear. So is there an opportunity to improve that informed consent between sponsors and patient advocacy groups? And then also where can regulatory and government agencies also help support that as well? Thank you.
We had a great, you were there too. There was a wonderful Duke Margolis Rise workshop for the Rare Disease Innovation Hub on data sharing. And it was a wonderful workshop, actually. It was about this. And I think the issue isn't so much consent. So it can be, but for all of our informed consents for all of our companies, we allow third-party data sharing. And so we ensure as a patient advocacy group that companies all kind of abide by that, because ultimately, if they are willing ever to share their data or their fluids or their samples, that we would be able to have access to that, and they don't have to reconsent the families. So we try to encourage that and advocate for that as a patient advocacy group and as a consortium. But it's up to the sponsor. It is not up to the consortium or the advocacy group unless it's just public humiliation, which isn't always wrong, but some companies will share. They really will, and they're amazing about it, and they feel it's their obligation to give back to the community. Other companies, it's too much money. They have a failed trial. Data sharing is not cheap. It's not free. And they are moving on to the next thing, particularly bigger companies. The smaller companies understand rare disease and the value of these samples and the data itself and the clinical data. And bigger companies may or may not. And there are examples where they do and examples where they don't. I think what's key for me is I think there should be an incentive. And I think the only incentive that actually probably has much value is an incentive by the regulatory authorities. And that incentive should be not a requirement, because it can't be, right? But it can be an incentive that if you share data from a failed trial or from a successful trial, placebo-controlled and sham-controlled data or baseline data, and you share that back to an advocacy group that has an appropriate data sharing forum, whether that's CPATH or NIH or whatever it is, then you get a priority review voucher of some sort, something similar, right? Where there's benefit to you. Whether you can sell it and make millions of dollars or hundreds of millions of dollars, that's probably going to dilute the value of a PRV. But maybe it's just a priority review. So you get 30 days for your next drug approval. I mean, an extra 6 months off your timeline or whatever it is, some incentive, it's either time or money. But I think that that's what will drive people to share data. And I think very few other things other than humiliation and just really good ethics will encourage people to share failed trial data. That's at least what we've seen. We've had some failed trials and very few people share the data.
I'd say from our experience specific to the ctDNA work that we've led, I think the companies that we've worked with have been very willing to share data. The challenge has been sort of finding the data that is, has all of the necessary characteristics in order for the appropriate characterization of that biomarker. But we've aggregated data from more than a dozen different companies at this point out of their willingness. And I think I agree with you. It comes down to sort of like that regulatory incentive to some extent. And I think in the ctDNA space, a lot of companies believe in the biological plausibility in the data so far that this could very well be a viable endpoint, but more evidence is needed. And in order to get to that more evidence is needed, there needs to be a degree of collaboration in order to get there, because no one company's portfolio is going to be substantial enough to be able to sort of complete the process here. So I've been very encouraged and impressed by the level of collaboration for all of the folks that we've had the opportunity to work with on that particular project.
That's great. We have Rob, did you have anything to add?
Well, maybe just add one thing. So candidly, I don't know exactly what our standard arrangements are in when we're consenting, right? So I'm not sure of how exactly we handle it. I mean, I will say as a company, we try to be very patient focused, and I think as part of that, it's maintaining a relationship with the patient and understanding the patient experience and journey. So, you know, when we're kicking off a process, for example, we're meeting with patients to understand what's impactful, and you're building a relationship with those groups with an intention that that's longstanding. So, I mean, I certainly think that comes into the thinking, and yeah, so I'd be out of beyond my limb if I said I know exactly what's in our clinical, you know, what's in our arrangements that we're reaching with sites. But I do think a focus on the patients and recognizing the patient as not just sort of you know, a disease, right? And a particular endpoint. It's recognizing you're dealing with a patient community and you have to understand what they're experiencing in order to understand how to develop your medicine. And ultimately, with the goal of not only getting to the market, but ultimately getting reimbursed and then adopted, right? Because if you go through and you get a great biomarker and you can get an FDA approval only to turn around and have payers unwilling to pay for it, or patients saying, well, you actually didn't deal with you did a great job moving this biomarker that FDA was interested in, but I'm not interested in that. It's not actually the thing that, you know, is affecting my life most acutely. And I wouldn't accept whatever side effects may come with medicine because of that. So I think it's a part of a general patient focus that includes sharing data as you can. And you know, I just don't know the specifics of how we approach that.
Yeah, and I think most companies start off there, but it's when they have a failed drug and they're moving on to something else, or a change in leadership in which we're dropping rare disease, we're moving to something different. It's too much money to go back and give the data to somebody else. So it's kind of like that rare disease is often cut off. Of like, we really enjoyed talking to you when we were developing the drug for you that we hope would be successful, but it's been a failure. So therefore, and some companies are not like that at all. They're wonderful, but others are. And it's very frustrating for the patients and families who put their kids through that and now don't have anything in return for the sacrifices that they've made. So it's really a challenge. But I think companies like Vertex are probably different than that. And other companies are very much that way. It's very company dependent that we've seen at least.
Great. One last question, which is from Slido, asks if, you know, as stakeholders seek to obtain early biomarker and endpoint feedback, are there any lessons that could be learned from existing pilot programs like the rare disease endpoint advancement pilot or the START pilot or things like that?
I mean, my understanding, I'm not part of any of them, is that the START pilot was fabulous, and particularly for rare disease and in CBER. And unfortunately, they're not taking any more applications for that. But my understanding was that was a perfect environment for that early and often feedback of we're having a challenge here, there's an adverse event here, there's a blip here. Can we get you on the phone and just have a conversation about what's happening in our early stage program? And my understanding from the Rett community was that's been an amazing experience. And we dream to have that opportunity because that to me is how you really develop drugs, particularly for rare, is to have, we don't know what's going to happen. This is first in human. We have no idea what to expect. And it may be off the charts amazing or really, you know, challenging with a lot of side effects. And so my understanding was that those relationships were wonderful and they go on, but I think there wasn't funding for them to do more. But I hope that they would consider doing more.
Any thoughts on the RDIA pilot?
I haven't done.
Maybe too soon to know.
We've not, but I think, I mean, in general, I think with the pilot programs, there's that intensive interaction is really what's hugely valuable. So, you know, I think not surprisingly, typically the industry perspective is that's great. So now do that everywhere else. And I think it is sort of like trying to.
DMC needs that. Everybody needs that. Yeah.
So I think you know, I think it is a question of how do you support the agency? How do you resource the agency? How do you do those things to have that kind of as much support as possible? But those sorts of intensive interactions, I think, are very beneficial. And then I guess maybe the other thing I would say with it is probably worth companies also taking a close look and saying, do you need all of those check-ins and what can you kind of create more efficiencies, recognizing there's a pretty constrained agency that you're working with. So.
I think the RDA pilot is, you know, sort of another example of the meeting type that's applicable to an IND situation. That was one of the requirements for entering that pilot is to have an active IND. So it also doesn't apply to the consortium model.
Great. Great. Well, I think thank you so much for the excellent discussion and for your contributions. We're going to take a 10-minute break.
Hey.
We're going to go ahead and get started with our final session of the day. So welcome back, everyone. If you don't know by now, I'm Valerie Parker, Assistant Research Director with Duke Margolis, and I'm moderating our last session entitled Refining the Roadmap, Priorities and Opportunities for Action. And so having covered biomarker evidence, infrastructure, development, and regulatory review throughout the day, this final session will serve as an opportunity to synthesize key themes, audience feedback, and areas of convergence or remaining uncertainty from the workshop discussions. And so by the end of this session, we hope to identify priority policy areas and actionable opportunities to strengthen the policy roadmap and support more transparent, consistent, and feasible biomarker use and regulatory review. And the team was gracious enough to summarize some key takeaways from throughout the day. So I'm going to begin with that, and we'll go from there. So the first takeaway that we identified was move from broad principles into a practical risk-based evidence framework. Participants repeatedly supported a framework that calibrates evidence to the biomarker's intended decision, context of use, prior evidence, and residual uncertainty. So not a universal checklist. A feasible first product could be something like a maturity matrix or toolkit with clear terminology, archetypes, and examples that support sponsor FDA communication. Another overarching theme that was identified was treat biomarker development as a shared infrastructure problem, not primarily an FDA problem. Scientific immaturity, fragmented studies, late assay planning, limited funding, and weak incentives often constrain progress before regulatory review. And so perhaps our roadmap should more clearly assign complementary roles to FDA, sponsors, consortia, patient groups, assay developers, researchers, neutral conveners, philanthropists, and government funders. The third theme identified was to prioritize perspective harmonization and evidence reuse. Reusable longitudinal evidence depends on aligning assays, metadata, collection timing, pre-processing, clinical workflows, consent, governance, and sample retention plans from the outset. Participants also emphasized balancing core standardization with flexibility for scientific advances and reducing burdens on patients, particularly in rare diseases. The next theme and final one that we've identified so far is make regulatory learning and engagement more structured and actionable. There was support for clearer public case examples, better communication of recurring evidentiary concerns, and standardized submission and feedback structures, while recognizing confidentiality and FDA capacity constraints. Existing meetings may address some product-specific questions, but consortia, patient groups, and small developers may need earlier focused cross-functional forums tied to concrete decisions. And so to facilitate our discussion today, I'm joined by our panelists here. So first we have Ramona Belfiore-Oshan, the Executive Director of the Duchenne Regulatory Science Consortium at the Critical Path Institute, who joined us earlier today. We also have Rasika Kalamegam, Head of U.S. Regulatory Policy and Intelligence at Genentech. And we thank you for joining our final discussion of the day. And then also returning, we have Isam Zaneh, Director of the Office of Clinical Pharmacology at the U.S. Food and Drug Administration. And so we do have one more Slido poll for the audience. But before I bring the whole room in, I wanted to turn to our panelists for just some initial reflections throughout the day. So Rasika, perhaps we could start with you and then from there go to Isam and then Ramona.
Well, thank you. Thank you, Valerie, and everybody. Thank you to Duke and to Bio and everybody who's contributed throughout the day. I'm really nervous because I don't want England to win. And I'm also grateful to all of you for sticking with me until the very end. I love Jude, but I can't stare at Harry Kane. That's on the record. It's fine. It's been a great discussion. And the fact that so many of you are still here, and I'm sure there are many online, is a testament to the fact that this is a really important issue for all of us to talk about and figure out a path forward. And as the day was progressing, I've been taking notes, and there are some reflections starting with panel 1 all the way to the end. So if you'll indulge me, I'm going to look at my notes. So just so I don't forget everything. The first one, which seems like an obvious, but is very true and worth repeating, is science is hard. I think Isam, you said that. It is hard. It's hard, and yet we all continue to invest in this space because while science is hard, disease is devastating. Yesterday, I met a former colleague of mine who is not yet 40, who shared with me that she's just been diagnosed with MS. I almost fell apart when I heard that. And those are the reasons why we are all in this room, why we are all united in trying to find better treatment options for patients. We heard a lot about rare disease patients and how difficult it is for them. I will say that is also true for diseases that have treatment options. You heard the story from Nicole about MRD. Well, why was that so important? Because we could have all as a community sat back and said, well, we have OS, we have PFS. Why do we need more endpoints? Well, we need more endpoints because we're continuing to strive to develop better therapies for patients. And when we're trying to develop better treatments, we want better measurement tools, better ways of assessing how a therapy will impact a patient without having to wait 10 years, 8 years, however long it takes to get that final readout. And that's why that investment is important. So that's number one. The second point I heard is we need to de-risk and get more predictable. I'm going to give you an analogy. De-risking is like wearing a seatbelt in your car. It's not the same as being predictable. Predictable is getting your GPS to tell you to take a route to get to a destination. Those two things are related, but they're not the same. I don't think it's fair to go to the agency and say, de-risk something for me and make it predictable. Because what sponsors want is a predictable answer, which means your drug is going to be approved, and they can never give you that answer, nor should they. So to your point, Isam, there was a lot where it was like FDA should do X, Y, Z, and I don't think some of that was reasonable or appropriate to ask the agency. De-risking is fine, but not predictable. Well, what can we ask agency to do? We can ask them to be predictable about process, about timelines. We can ask them to say, give us meetings in a particular cadence, in a particular timeline. That's fair, but not tell me that if I do XYZ step, you will approve my drug, because I don't think that that's fair. The last thing I'll say is collaboration is important, but it's also important to understand what each collaborator can reasonably be expected to contribute to the best of their ability. It is not fair to expect FDA to be a university or an academic research institution. It is not fair to expect a patient group to become a drug developer. Those are very different endeavors. Yes, the community needs to come together and work together. So one outcome of this meeting and this proposal might be to refine things and say, what can we reasonably expect from each collaborator so that together we are finding a path forward? And then the last thing I'll say is there was a lot of discussion about sharing information, data reuse, et cetera, et cetera. As sponsors, we don't compete on a biomarker or an endpoint. A biomarker is a biomarker is a biomarker. It is available for everyone to use. It's scientific knowledge. It's out there in the commons. We compete on assets. EGFR is not owned by anyone. It is applicable to everybody. ALK is not owned by a sponsor. It's available to everyone to use. So let's be clear on what do we mean when we say data use and reuse and knowledge sharing, because there is a lot of knowledge that's already shared. Which brings me to two other concepts. One is we talk about the totality of data, the totality of evidence. We need to also get familiar with another concept, which is weight of evidence. It's not just what evidence is out there, but it's how are you weighing each of those in that particular context of use. It's a concept that I think Isam is deeply familiar with, and I will let him speak to it. And then the very last thing I'll say, which is a very tangible ask of the FDA. Sponsors, when they are using a novel biomarker or novel endpoint, are usually de-risking the use of these things by launching it not for the first approval of a drug, but the second, third, and fourth approval, because your drug is already out there. And now when you're trying to figure out what else you can use it for, you're able to take a greater risk and imbue a novel endpoint or a new biomarker in your study. So what is my ask? FDA is great about putting out the SBA for the first approval. It comes out within a very regular time frame. But the second, third, and fourth efficacy supplements, they can take forever. They can take years. It's a very simple and tangible ask. Can we get those efficacy supplements released in a timely manner? Because that's where we will know, what did FDA do with that novel endpoint? What did they do with that novel biomarker? I'll stop there.
Thanks so much, Rasika. And Isam, just before your comments, I just wanted to pull on a few threads there. So first, really appreciate you highlighting, what is reasonable for each stakeholder. We know that we have a lot, and this is our preliminary roadmap in there for FDA. And we certainly appreciate all the feedback that we've gathered throughout the day, and promise you that won't be in vain, because our goal is exactly what you also mentioned, Rasika, to really refine our roadmap and to do a prioritization exercise to really think through what is reasonable for each stakeholder group, because that's an important point too. The other session I moderated was regarding collaboration. And so if we want everyone to come to the table, what are those roles? Like perhaps, I do see patient groups venturing more and more into the drug development space or into more of the research space, but what is everyone willing to do? What is a reasonable expectation for these groups, for FDA, so that we can all work towards that shared goal? I really just appreciate you highlighting those points. But Isam, please, what are your reflections for today?
Maybe I'll respond to your themes that you laid out. I think the team did a nice job summarizing them. To me, a kind of evidentiary framework or risk-based credibility framework seems like a no-brainer. That's come up several times today. Easier said than done. One thing that came to mind was we have consortia for a lot of different diseases and modalities, but there are industry consortia that could take this on. Right? Why not have a consortium that works on an evidence framework? And I think Pujita and her team have done some fine preliminary work. I would say also we may not have to reinvent the wheel on this. ICH M15, which just came out recently, is an evidence assessment. It's a risk-based credibility framework. It's model-informed drug development, but it's completely portable. In fact, we stole it from the engineers. And so the environmental toxicologists, they don't test. They don't confirm hypotheses that a substance is going to kill you, right? They use in silico modeling and non-clinical data. So there are all kinds of sectors that we can sort of borrow and synthesize this idea of what are the dimensions and domains of credibility around a biomarker, depending on what we're hoping to use it for. So I think that would be a worthwhile consortial effort. The second piece, and I'm going to kind of clump them together, is this shared problem and this kind of like reuse. So the biomarker ecosystem. I heard a lot about incentivizing. That is uncharted territory, I think. We obviously can't lobby at FDA to incentivize, but there's no 5K for biomarkers, right? We have 5K runs and marathons for our favorite diseases of interest, but we don't have biomarkers don't sort of capture that space, occupy that space in the imagination. And to the extent that many of the people in this room, whether it's patient advocacy groups, pharma, bio, and so forth, have some cachet on the hill, it might make some sense to start to raise the awareness of the importance of biomarkers in this entire ecosystem. Biomarkers are important to the diseases that patients and clinicians care about. And so that might be something to really think about. What do those novel incentive structures look like? It's not within our purview at FDA to incentivize data sharing, but that is something that could be thought through a little bit more and, be instantiated through statute and regulations. The issue of reuse and resharing, I mean, this is very interesting. I feel like some companies just do that better than others, and that also could be a consortium effort too. Going back to the point I made earlier about the early days of genomics, where we were trying to ensure that within a development program, there was sufficient sample acquisition, informed consent in order to facilitate biomarker discovery and validation in the future. That seems like, we do have actually a lot of lessons learned that companies could be sharing, again, through maybe perhaps a consortium effort. That to me feels like a rich opportunity. And then how do you leverage that? How do you leverage open science? There's so many. I mean, there's smart high school kids out there solving problems with computational power. I mean, it's like incredible the amount of brain power that's being brought to bear on what used to be intractable problems. So, are there ways to think about more, open science in terms of biomarker, not just discovery but validation? That includes, some of these dimensions of some of these evidentiary dimensions that we would develop under a framework. And then the final piece is around this regulatory learnings and engagement, and I'm going to separate engagement and learnings. The last session was really interesting and eye-opening, and we do have a bandwidth problem. I think from my experience at FDA staff across divisions and therapeutic areas are more than willing to engage with patient groups and with companies on these issues. It truly is an issue of bandwidth. I mean, if a company has a half dozen neuroscience assets or, one rare disease asset, have to remember that FDA has all of those assets. We're responsible for the evaluation of everything that comes through the door. So, multiply your workload issues, so to speak, by, all of the molecules, all of the programs that we see. And it's a really tough thing to be involved in product development and facilitating sort of the science that underpins, robust biomarkers. It might be worth looking at all of the various types of meeting mechanisms that we do have at FDA, including non-regulatory, like the critical path innovation meetings, and maybe some of these others, and then to begin to map out what meetings would be most appropriate for what kinds of biomarker questions. And if there truly is a need for a new kind of meeting, that might be worth considering. If we can already leverage the stakeholder engagement mechanisms that we have, all the better. On the learnings piece, to me, this seems like one of the stickiest issues because, I've heard several times we'll just release the summary basis of the supplementary approvals. I mean, the redaction takes forever. It really does. All of the supplements that we have to redact in order to make sure that confidential and proprietary information is not released. But even if we had all of the bandwidth to release those, you may be surprised to find that that's not where the biomarker conversations happen. They happen in the IND space, right? That's where it happens. It happens in the meetings with companies. It happens in all of these, type A, B, C, D meetings, interact, and so forth. And that's not synthesized all the way to the end. There's, that might be invisible by the time a drug gets approved. And it's certainly not visible when a drug doesn't get approved. So I would actually consider shifting that responsibility to companies. I think companies are in a very good position to synthesize, well, here's how we dealt with uncertainty around this animal model that didn't translate, or this evidence that was incongruent, and this is how we sort of overcame it, or this was our conversation with FDA, and this is how we got from a no to a yes, or from a yes to a no. That would also be helpful. That I think would be really important. Development takes a very long time. So if we're waiting for approvals and the summary basis for approvals, there's a lot of space and time that I think we could be leveraging better. We also have surrogate endpoint resources tables. We have the Leader 3D. I think Pujita mentioned those cases. So we do put cases out there that are intended to be, broadly, they're specific but broadly illustrative. So I would encourage, maybe an action item is to take a look at the universe of resources that not just FDA, but EMA, PMDA, other regulators globally put out there that try to speak to this question of biomarker science, and then see, okay, well, are there gaps that then we can begin to fill, either from a knowledge management and dissemination standpoint or an engagement standpoint. And I'll leave it at that for now.
Thank you so much, Isam, and thank you for providing an FDA perspective throughout the day and helping us to refine our recommendations. So many thanks there. Ramona, what are your reflections?
Thank you. And it is really hard to go after these remarks. And I'm sorry, Rithika, for the scores. As we learned, there is a thumb down for that.
I know.
But really going into the overarching learnings from today, I think one thing that resigns really that the discussion is improving biomarker use and developing biomarkers requires much more than generating the evidence. It perhaps goes more into understanding as a community level what evidence needs to be developed, who is responsible for developing this evidence, and how the evidence matures over time, and how we can be also responsible for addressing that maturity, and how we can move from one context to the other. Looking at assessing the reuse of this data is really trying to maybe look at commonalities across larger communities rather than focusing on the single targets, so that we can speed up the process of development of biomarkers and have a high impact, and then stratify and extrapolate for specific case studies. But in the meantime, moving forward, specifically in the rare disease space where there is such a high unmet need with so few numbers of patients that can participate to trials, I think we need to find solutions that are more driven by common downstream efforts that can be then reassigned back to the single disease pathways. And so work maybe on pilots that are more family of disease oriented rather than the specific disease, and then align on the requirements for the evidence needed to get to the packages and the elements that really go into the assessing the development and the reuse of this data. Certainly, as a component of the Critical Path Institute, there is a fundamental advancing knowledge into the power of a public partnership and understanding how we can come together to reduce the burden for single sponsor investment in biomarker development. This is certainly not something that the FDA plays a critical role on in the development space, but many of these solutions really require more of a ecosystem challenge. And we all should take this challenge, and agree very much with the point that we put so much on the patient community in advocating for the development of biomarkers, and maybe also addressing some of the challenges for data sharing. But each of us should really be responsible for advocating for this process to happen and enforcing those processes at all levels, going from those projects they are granted through NIH funding, FDA funding, and going through enforcing the data sharing following that actual grant approval, and making sure that there is a return of this data back to the community. A few points that I think are relevant is really going into adoption of the technologies. I hear building communities and coming together in working groups all of those things do exist already. And what we need to make sure that happen is that we actually adopt them. And sometimes we fall so much into the wanting to reinvent the wheel, but it sounds like we can rather go back and inform each other more quickly onto what's already available out there and how do we get to the adoption of certain tools, like was mentioned, model-informed drug development methodologies, AI, machine learning-driven tools. We have the technology for advancing NAMs and the simulation in silico of some of these scenarios without really burdening the patient population at early on in the development stages. So it's more about how do we incentivize this community that, especially in the rare disease case, is composed by parents and patients that are driven by really the unmet need, a really urgent time frame that might not be expert in those technologies. So we need to fasten that communication in supporting the patient community in both leading the efforts, but also giving them the right tools. Because again, we have the technology, we should push through the adoption piece more than the reinventing the wheel.
Well, thank you so much to all our panelists for these really thoughtful remarks. It's been such a great day hearing all these different feedbacks and components and how we can modify our roadmap. We are running short on time, so I just want to do like a lightning fire round, and I'm going to give you 2 questions to choose from. So perhaps a bit unusual, but taking moderator liberties. So the first question would be, which 2 or 3 actions should be elevated as our roadmap's highest near-term priorities? That's the first question you could answer in the lightning round. The other option would be, what could realistically begin within 12 months? And then what measurable progress should be expected within 5 years? So a much more kind of timing-oriented one, or what are just 2 or 3 actions? So perhaps, Rasika, we can start with you and then go in the same order.
Well, I'm in a funk because somebody just signaled to me that England scored.
I'm sorry. Well, my moderator iPad doesn't get
I'm in a funk.
scores.
Well, I'll take the, what are 2 or 3 action items? For me, I think we've all been part of these consortia. We've all been part of these conversations. And Isam, you were talking about FDA Can't Advocate. I'll just say that's true. But 10 years ago, the patient community, and I worked for a nonprofit group back then, we were the ones who went to Congress and got it written into 21st Century Cures. I mean, this whole concept of qualification came about because of advocacy, which was already done. And here we are 10 years later, we're taking a look back and saying, did it work or did it not work? And this is the refinement, the next step of how do we get better at it. So an action item coming from today's discussion for me would be the stakeholder community is well versed with the problem. They all know what the issues are and they're all trying to solve it. What is an effective way for each stakeholder to contribute to this problem where they're not duplicating efforts, where they're staying within their expertise and their remit and contributing to this? That to me would be a huge win.
Thanks so much. Isam?
Yeah, I think I've sort of spoken to this question already throughout the day, and I agree with that. Maybe I'll just add, as you go back to look at the roadmap, maybe thinking through what has been done, what are we proposing to do differently? That to me came to mind when I was hearing the conversation around public-private partnership, and that is, what would be a transformative gesture in that space? And I don't know the answer to that. The other is, this is my personal opinion. I should have put the disclaimer out there. I would really look at the things that if as a community we have not solved by now, I don't think looking to like any one entity, whether it's FDA or whatever, to answer that question is going to be helpful. The one that came to mind for me was this, like somehow articulating what clinical meaningfulness means. Yeah, that to me does not seem a worthwhile effort. That's completely subjective. It's much more, I think, useful to come up with that set of considerations that facilitates the dialogue, as opposed to sort of quantifying, like coming up with like a binary answer on something like that. So maybe just going through that as well might be helpful.
Thanks, Isam. And Ramona?
Yeah, well, addressing some consideration, what can we do different? Invest more. Investment needs to growth, and there needs to be much more investment ahead of the planning, not as an afterthought. Biomarker development cannot be assessed after the data has been collected already. We need to work on the anticipation, establishing those alignment across protocols and validation, pre-analytical, legal entities challenges. How do we make sure that if sponsors are trying to adopt these tools across multiple different regions, can comfortably do so and be motivated and incentivized in investing in biomarker development? So there needs to be a whole restructuring of how we're thinking of biomarkers as an element for addressing drug development tools that really comes ahead of time rather than as an afterthought, and invest in that as a fundamental piece of our drug development process. That would be it.
Thanks so much, Ramona. And with that, we'll have to leave it there. If we could have a round of applause for our panel. I have a few closing remarks, but and please feel free to leave the stage. I'm going to stand up and give our closing remarks, but thank you so much for joining us today. And I know we're a little over, so I will make this brief. But before we close today, I wanted to thank today's presenters, moderators, and panelists for sharing their expertise and engaging so thoughtfully with our preliminary policy roadmap. I also wanted to recognize the Duke Margolis project team, Nicholas Harrison, Patrick Rodriguez, Maddie Cordell, Rachel Hendricks Stirrup, as well as our events team at Duke Margolis, Luke DeRocher, and Hannah Vitelio, as well as our other colleagues who supported the research, planning, and execution of today's workshop. We're grateful to the Convene team here for hosting us today, and to our bio workgroup colleagues, including Pujita Vaidya, Darcy Freer, Jewel Martin, Lauren Olivia, Cecilia Yoder, Steve Berman, Annetta Beauregard, Snehal Naik, and Allison Kobe for their continued partnership and guidance. We also sincerely thank the many stakeholders who contributed to this research through interviews, feedback sessions, expert discussions, and review of earlier versions of the preliminary policy roadmap. Their perspectives have helped shape both the current version of the roadmap as well as today's discussion. Following the workshop, we'll review the commentary, survey responses, and ideas shared today as we refine and prioritize the policy recommendations. I also wanted to announce today that when we distribute the recording of this workshop, we'll also have a survey released about our roadmap to capture any additional, more detailed feedback you'd like to share with us. That survey will be available on our event webpage and as well as in the email. And then findings from that survey will also help us inform revisions to our recommendations. And so just thank you again for joining us today. Appreciate all of your time, candor, participation, and yes, that's you in the audience participating in our poll. Thank you. Have a good day. I hope that the game isn't over if you're trying to catch one last glimpse. Take care.